bottom. One is to sample a sulfide into an RNA-DNA preservative in one of the coral quivers. Next, we'll head over to a diffuse flow at this basalt crack, which should be a treat. It's apparently a crack large enough to get the vehicle into. And then, if we have the extra bottom time, they probably can't hear me because we're restarting the stream. No, never mind. Oh. Then, if we have the time, we'll take some more super samples at this smaller chimlet with flanges. And then we'll bring Sebastian home. We've had to retrace some of our steps this dive because we weren't sure the super was going to be up to the task of collecting in situ preserved and non-preserved samples for all of our sampling points, but it seems to have been behaving most of the dive, so we're going back to get those paired preserved samples.
So you want the super on or off?
You can leave it on.
It's going to be like I think like an 8 to 10 minute transit to the next site.
You know where we're going, but
We're not leaving yet.
We're not leaving yet.
we're not going anywhere. So
yeah,
that's how good.
We have to grab a sulfide.
I just wanted to update you on how long it was going to take.
Okay.
Yeah.
Should be okay.
Do you want to pop in here?
I can point to a rock. That's fine. Yeah.
PhD in pointing to rocks.
Yeah, that's what I got my PhD in, pointing at rocks. Go in here.
Just a heads up, your station keeper is still on.
If you would feel more comfortable turning it off, we can.
No, he's
Oh.
just.
Oh.
So if he's gonna land out on his ramp, he's just gonna turn that off.
I see. Okay.
It's just a different.
Never mind. This one.
We have two primary objectives and one secondary objective left. We're sampling the sulfide, then we'll transit 7 to 10 minutes, do some super sampling at a diverse site, at a diffuse flow site in a basalt crack. After that, if we can, we'll do some super sampling on a chimlet, and the plan is to be off bottom in a bit over an hour. After that, we'll stream as much of the ascent as we can and try to see some sights.
Cocoa in your tea?
Oh no, it's mud. It's mud.
Oh.
Mud mixture. No. It's grown up.
Grown up hot chocolate.
Yeah.
Where's the peppermint schnapps?
Yeah, you should be able to reach them. If not, you can still swing it a little bit. Sure. What am I doing to it?
You're grabbing it. You're grabbing it.
Okay.
6 reads is free.
Yeah,
Brought it up yesterday.
yeah,
So you gotta break up for it.
yeah.
Okay. Cool.
Right, Coral Quiver 6?
But Core Cover 6 reads is free on my map.
Yep. Well, and that's what it also says in the objectives.
And the new objectives?
No, they're always their objectives. Coral Quiver 6.
Yep.
Was it not enough yesterday?
Well, yesterday, none was put in the Coral Quiver.
Oh, it was all in the bylaws.
It all went to the bio box. Yeah. Yep.
We actually do have multiple fissure markers, so I am really not sure this is the Sebastian-sized crack. That might be fissure 1 rather than basalt crack 1. Wasn't on watch when they dropped it. Good morning to those who have just joined us. Thanks for tuning in. You're catching the last couple of hours of our final dive on the Natividad vent field on the Western Galapagos Spreading Center. We're sampling a high temperature sulfide into RNA-DNA preservative right now. We'll go on to pump a few more superfilters, and then we'll head on up. And Josh has been so kind as to stream most of our ascent for you. We have, so our sort of invertebrate specialist, Nicole, has a couple of those anemones of that species, though the ones that she got are from mussels. They're incidental collections. We're somewhat constrained in our sampling options by the permits. We have a permit that covers siphonophores, but not anthozoans. The anemones, I agree, are pretty cool. Don't generally see them this close to hot flows.
Yeah, we'll take it and then we'll go back in.
Ooh, glittery.
Very glittery. Oh. Neat.
Which you
Rachel, is that the half-strength stuff?
Yes.
Ah, thought it looked nice and pink.
Which is then you'll be back below, you said.
Yeah.
So I should open up the flow further down there.
Seeing it.
So that if it's where
Yeah.
we're probably gonna hopefully be breaking into the flow further down.
beautiful. Look at that.
Oh my gosh. Observation.
You're gonna have to tamp that one down.
There it is.
Tamp it.
It's pretty veggie-licious.
It sure is.
Are you okay with that?
Yeah.
What size would you say that is, Rachel? You always give such great size descriptions.
American football.
American. Thank you.
There's some
I
scale
don't know what
marks
league
on it.
regulation that is.
Tailworms, okay.
We did mix our own RNA later, but there's some interesting complexities associated with making RNA later at 30 or 40 degrees Celsius. RNA later, by the way, is a saturated solution of ammonium sulfate and a few other salts. We make it at 30 or 40 Celsius, and then we send it down to the bottom of the ocean where it's 2 Celsius. Anybody who's ever tried cooling salt water knows that the solubility of the salt changes.
Yeah.
We just crush it down in there.
Yeah,
Tamp it.
Yeah, just tamp it.
Conceptually speaking, it's a little bit like putting a football-sized rock into a baseball-sized quiver. You put all that salt in there, and then you cool the solution, and it loses its ability to hold the salt in solution. So a lot of it crashes out. For that reason, and some others, this RNA later solution was mixed up with half the absolute amount of salt, but it's probably still pretty close to a saturated solution at this low temperature.
You might need a minute to pull it back out, but get up on the surface. Needs Mike.
Well, the RNA later's working. That worm looks pretty preserved.
I don't know.
Let's see.
Now you can stamp on it. Yeah.
Which rock number is that?
Which rock?
Which rock number?
R08.
R08, sweet.
R08.
Most
What
excellent.
a dive.
The best collection of days.
That's
It's
not
a little bit old.
still a little bit wonky. I mean,
You
'Cause
want to
the-
keep the RNA later.
yeah,
That looks good.
it looks pretty good. I mean, the RNA later is more dense.
If you don't mind calling out the timestamp when we...
Most of it should stay in
So you want to leave this?
there. No.
Yeah, now we're going for rugby ball.
Right. Beach ball. The whole chimney.
Oh, that's right. You pushed us up.
All right. Excellent. So we can now move to the diffuse flow here.
That's basalt crack 1.
So crack. Okay. Cool.
And do you mind calling out a timestamp for the R08, please?
Yes,
We are
of
going
course.
to sulfur
That
one, which
is
is right behind you.
102245.
No, that's right.
10,
I
22.
gotta have to
2,
it is
4,
right
Thanks, Haley.
here.
10,
So it's 70 meters and a bearing
Um,
of 90 degrees.
Oh no, you're gonna
No,
be over
I
there.
gotta-
forever.
I gotta
That's
do
what
another
you-
RNA
I'm just
later.
gonna
Well,
stay here the entire shift.
While we're in transit, Haley,
Yes.
do you mind either you can do it or you can pass it over, but I think it would be nice to try to get eyes on IGT 3 or 4
Okay.
so we can see what that sampling locale
Yep.
looks like.
I'll look it up here.
The crack seems like it could be, could provide a lot of options.
No problem. Review.
Great. Yeah, go ahead. We have to move them a little ways to the north.
Okay.
Thank you much.
Gosh, it's just nothing that's consistent. Noted. Okay. Try. diffuse. You want to try to search function or something?
Yeah.
Because
It's not exactly
it doesn't work
user-friendly. No.
Yeah. Well, we're also inconsistent with our.
Hmm. Yeah, that helps.
Can I push this onto the screen? Yeah, no, that's the way. I don't know if you can. You can move that 1 point though.
RO8B smushing. RO8B smushed.
Yes. Very efficient.
Collection. Glittery.
You're welcome.
You
You can make comments on it if you decide.
Oh, I appreciate it. Perfect.
So what was that? Use the go-to.
I don't know. Rock. Got one of
Wait,
them.
yeah, you can use go-to. No, but I don't think it was, which is fine, I just want to make sure you can see where it's going. Which is fine, manually putting in.
Okay, I think I found it.
Found it. Thank you. That
Think
feels like
I
Jacob
found it.
manifested.
It's a
Thank you.
toss-up.
And if it was a text, it was a document, I can search.
Yeah.
The newer versions have it.
Yeah, newer emergency log.
Mm-hmm.
That's good to
The
know.
ones we're working on. Really.
Mm-hmm.
Alright, there you go. So we
Has
have
good bones, the software.
Right.
Um,
The handover. I got the vessels on.
30128.
Where's this sound? There it is. How do you tell which microphones are on? Up there. So if the knobs aren't on 0, they're on. This is the last one. Last one's right here. And the back one is right here. It's one. And all of it's one. So where, we'll go to the rest of the line. So you said this starts all the way up there. That will just right back. And then this is, and then on. This one, turn it down until it cracks up. Oh, as you're ready. That's what it's with here. That's what makes it right there. I don't know. It's like clear cross. Oh, really? Yeah. Oh, okay. So that's good.
It has some serious problems.
You're telling me, right?
I'm gonna use my phone instead.
Right. Serious problems is right.
I got the timestamp.
You got the timestamp?
Yeah.
Nice. Yeah, it's
I like how the after date starts at January 1, 2000.
Yes.
At least it's not 1970.
Yeah, that's a good.
Could be worse.
Yeah, absolutely. Honestly, I'm tempted to just download the events. Download.
You see, probably. school that went over the 90s. Before they're engineering school. Yeah, it's all. Can't do anything right. Yeah.
The fissure, the crack. Stand by. I'm trying to get some video of it from before.
Jim, what am I doing, Jacob? Here. Do you want this?
That's helpful.
Wow.
Thank you.
Yeah.
Higher agency. The temperature is helpful.
Is it in there?
Temperature, same diffuse flow.
And you won't see the
Near-
temperature anomaly
yeah.
until you get down there.
Mussels one near Chimlet 1 equals basalt crack 1. So yeah, head for this marker, basalt crack 1. It should be pretty specific.
Nice.
Heading of 015. Yes, just east of north. And maybe you take us, if we trust the tracking, maybe you take us 2 meters to the south. You could just thrust
Oh,
out.
there, right there.
Yeah?
Yeah.
That, yeah, kind of works.
That right here.
Yeah? All right.
We'll have to go down a little bit closer. You want to zoom in a little bit?
I'm gonna wait for Ben to steady up.
It's either that or that. There's more animals over here, so maybe it's
Yeah. So I was here. Let's go back to part 4.
yeah.
It's further south on the tracking. We just have to see how well it's held. Hang on. It's drifting?
I don't think it.
Okay. Good. This was only about 7 hours ago that they were here.
I mean, it would be right here. Sort of. I mean, I would need to get down closer to see if there's a diffuse. I mean, there probably is a diffuse flow given all these animals. There's a lone mussel right here, which makes me think that it could be in here. Seems large, kind of shimmery.
Unfortunately, this is missing from the stream.
Yeah. Yeah, I think like right in here is where we can set down. Nope.
Oh.
No solicitors. Yeah, if you want to put it right next to that little mussel, the super wand and the pH probe, I think that'll be useful for multiple parties.
How tightly paired do you need this to be with the IGT?
This is gonna be the same
Cool.
area. It'll be the same chemistry.
Yeah.
And doing this gives Roxy a chance to find symbionts.
Cool.
We're doing super.
We are doing super.
And pH probe for now.
Yeah, pH probe and super wand. Yeah.
Thanks for this.
Of course.
Right
It's
above.
marginally helpful. I mean, the reference system is still totally
Yeah.
required, but that is helpful.
Yeah.
And I'm glad it's not that critical.
Yeah.
Landscape.
Thank goodness.
No substitute for paper.
Yeah.
Morning.
Good morning.
We're gonna do super over this diffuse flow, so there's this one little lone muscle. Cool. Great.
There might have been more yesterday.
I don't think we sampled the same.
No, I don't think so.
No more rocks. We have collected all the rocks. Yep.
Some salt. Is that low enough for me to put?
Try to get a little bit closer.
Okay. I'm not touching it.
Don't touch it. No touchy. That's perfect.
It's an interesting little worm. Looks like a
The one that's floating by.
yeah. It's acting like a floater, but it's awfully close to the bottom. Oh.
Can you come further in on the first chance? Might clear on your high temp probe. I might be smashing that too then. No, just step up this one there on that side.
Let me just point it a little bit.
That it? Seven. No. Don't worry about that. Bracket.
Better view.
Okay.
You want to grab a good picture on the side. And you got zoom on the side.
That looks great.
Just do the trees. Yep.
Hold there. Right here on IS. Urge, 120 seconds, starting now.
Can we drop a marker here, please? Super diffuse flow.
That's so weird because this doesn't change scope. So it feels super wrong. So here, that's probably the better one, but you know what I mean? Because it's, that makes sense.
Serpulid.
Circulid. S.
ERPULID.
Super or super?
Super is in suspended particle.
SUPR.
PR. Yeah.
I don't know if you're describing the state of the flow also.
No no hyphen.
That would make sense.
Hey, Jacob,
Mm-hmm.
what else is on our list today?
After this, if we have the time, to super the pagoda, and that's it.
That's it, okay, great, good, yeah, we should have time for that.
I know that we loaded the super sampler with 14 cassettes, but in the master sheet, there's only 11 listed. I don't know. Okay.
There's a few more muscles in the crack there. You mind if I play a little bit?
I was there for the loading.
Maybe they just didn't get already.
They just didn't fully populate.
Okay.
Yeah, I would. Yeah. Okay, purge complete. 10.
Oh, no, to each other.
Beginning 700 seconds, pumping
Oh, she's okay.
now.
If it's off, it's just not far out.
Good morning. I don't know if that's a compliment to me or our MT Josh, who is actually handling the audio, but it's a team effort all around here, and we appreciate you tuning in. We're taking our kind of our second to last super sampling campaign of the day. Zero, 5, 1, 0, 3. For those of you who recently signed on, welcome. You are diving the Western Galapagos Spreading Center with Sebastian off the Falcor 2. And you're in the control room with the Dawn Watch 4 to 8 a.m. Donner Party. That's the DAWNER party. Haley, Rachel, and Jacob.
the name of, was this IGT 3 and 4? Diffuse?
I mean, yes, it would be paired with that. Nice bunch of mussels here.
Yeah, like they're underneath the rock.
Yeah. So there's the one to the left of your sampling head, and then there's,
Will you note that, Rachel?
Yeah.
That'd be important for the data.
yeah,
Thank you.
several clumps of mussels.
We're wanting to be offloaded by 5:30 or 6, yeah.
Oh,
I think 6 is fine.
plenty of time.
Because, then I guess it's that fun day right after breakfast. I was gonna remember when that.
Check out these serpulid featherworms. These are one of the main kind of ecological hallmarks of this site. We did not see nearly so many at the iguanas or pinguinos fields, but they are all over the place here. And it also looks like, you think those are zoanthids or proper anemones? Anyone?
Look at zoanthids.
Yeah, would be my hunch.
I mean, we've definitely seen anemones here, which is wild.
Mm-hmm.
Right on the
Yeah.
fence. Did Nicole say, isn't she the anemone expert?
She has a couple specimens. I haven't gotten an ID from her.
But does she have a hunch as to how they can handle the sulfide?
I haven't asked her, but I should.
They don't like they're typically in very high sulfide.
Well, that's the
Well,
interesting thing.
At the one chimney where we just were,
Yeah.
there was like a couple right next to the orifice.
But even that, like, it doesn't necessarily mean they're in high sulfide because the fluid is buoyant. Depends on how permeable the chimney itself is.
So right now, Dr. Roxanne Beinart and Dr. Rachel, there in the back of the control room, are discussing how in the world the anemones, which to be clear are not the zoanthids we're presently looking at, but the small kind of white, off-white anemones that we've been seeing on vent structures, deal with the sulfides coming out of those vents and whether they indeed are exposed to high sulfide at a small scale. The autofocus is doing it again.
It's not autofocus.
When I turn the dial, it kicks back into auto. Yeah, we were discussing the anemones that we've been seeing on some of these vent structures. To be clear, those aren't the zoanthids that we're looking at right now, which are at a diffuse flow site, but closer to some of the high temperature vents where we've been sampling, we've seen these little white to off-white sea anemones. And we were just discussing how much sulfide they actually get exposed to, because obviously it dissipates very quickly after it comes out of the vents. Anemones and other cnidarians aren't generally known for being able to deal with high amounts of sulfide because they have an open circulatory system that basically just contains seawater. They don't have blood or proteins in it that can help to sequester that toxic chemical. Generally hydrogen sulfide, yes, though it can rapidly bind with transition metals that come out of the vents to make compounds like iron sulfide, which are the main constituents of black smoke where we see it. Hydrogen sulfide, though, is membrane permeable, so it's pretty acutely toxic. It has about half the acute toxicity, at least in the typical test organisms, of hydrogen cyanide, but it's not generally treated as such a dangerous chemical in sort of industrial and laboratory settings, because unlike hydrogen cyanide, hydrogen sulfide smells really foul and will be totally unbearable before it reaches dangerous levels. It sort of announces its presence. Not just another worm in the crack being dramatic. Yeah, these are in a sense tube worms. They make tubes, but they belong to, I believe, the family or order Serpulidae rather than Ciboglynidae, which is what the giant tube worms like Riftia or its smaller cousin, the Jericho worm Tevnia. They are Ciboglynids. These are Serpulids. They don't have the red plumes, and they're a lot featherier looking. Also, they really dominate this site, so if we come wide, you'll see that on these diffuse flow basalt beds is just worms as far as the eye can see. Serpulid worms. Serpulid worms, squat lobsters, and these bony-eared fishes. Kind of the order of the day at Natividad Vent Field.
Yeah, that was what I was attempting.
We've also found some really nice sized mussels at this vent site, though. You can see a few nice clusters of them right on the diffuse flow site we're sampling right now. We collected about 25, give or take, earlier on this dive. And those are going to go towards genetic connectivity studies being done by Dr. Corey, some incubations being done by Dr. Andrea. We're actually going to take some of these into a special container we have on deck that's set up to hold deep sea vent animals at their native pressures. So we'll keep them under the same physical and chemical conditions that you see here at the vents for a few days and monitor their physiology. Ooh, those are tiny Riftia in the back.
Really?
Yep, little babies. Hang on, let me get this thing out of autofocus.
Oh yeah.
Directed towards the machine, not any person in particular.
I want to focus for them.
You see them?
Yeah. We found Riftia.
We found a couple of others at this site.
Yeah,
They're
we've seen this on a couple others.
roughly the same size as I recall.
I wasn't there on the 7, but yes, I said small. So. Rachel, wait a second. I'll put that in the super long too.
What's our lighting scene? Will the lowers get there?
You like look at that?
Lowers
Yeah, that's
washing
fine.
up.
Oh, okay.
Just turn the lowers off.
Thank you.
So do you want them, that's all on?
No, whatever you think is best. downlight would be ideal.
So that's all the upper slash down.
Okay, cool. That's great.
So this.
We can fix the white balance later.
There you go.
Yeah, I'm mostly concerned with focusing on them.
Can you focus now?
Can I focus now? Let's find out.
There you go.
Yeah, it's tracking.
Nudge.
I'm gonna try to center up on them and get some sizes. We gotta get the lasers back there. Okay, okay. Ah. Hold.
Good.
All right. If you don't mind
Got it.
observing lasers, Riftia.
Yeah.
So the little one looks like from the base of where we can see is maybe 8 centimeters. The bigger one, if we were to scale it up, might be more on the order of 12 or 15. How's our
It just completed.
fantastic?
So, let me switch over to RNA.
Okay.
Okay, pumping for 15 seconds.
Could we lasers off, please?
Turn it down.
If you want, we can do a macro highlight squat lobster or something like that. And Riftia, it's up to you. This isn't exactly high value stock footage.
Okay, pumping complete.
Yeah, so both the Riftia and the mussels have bacterial symbionts. I'd rather have Dr. Roxanne pitch in on those since she knows a lot more about them. But what I can tell you about the way the animals make a living is that the Riftia depend entirely upon the symbionts for their nutrition, whereas the mussels can filter feed if absolutely necessary. And that's one of the hypotheses that I've heard floating around the dining room here about why we've seen plenty of mussels, a few clams, but very few Riftia on this vent system, is that the sulfide fluxes may just not be quite high enough to support large amounts of worms right now.
Also heard Pete talking yesterday about the temperature not being warm enough because the Riftia prefer like around 30.
So if you, I'm not sure whether you all can hear Dr. Rachel back there, but she's also suggested that it might be a question of temperature tolerance. The Riftia like it a bit warmer. The mussels can tolerate colder, and again, with weaker flow come lower temperatures.
Do we want to put the pH probe right over the Riftia just so we can get a temperature and pH just to log it?
It seems like a cool idea. We just
You
need
can
to
get it back there, I don't know how.
make sure we get a timestamp on that.
We're gonna have to.
We'd have to move the vehicle.
Zoom out and
Yeah,
see how
we're
far we are.
finished
Make
with
sure we're
supering.
far.
We're,
So the super wand can go.
are we completely finished with supering?
Well, if we have time, we can go to the pagoda and sample there. But for this site, the specific sample, we are finished.
Great. Okay. For some reason, I thought you had to take a second filter here. Yeah, if we're gonna pick up, it would be nice to get the pH probe into that crack and get a couple of temperature data points, pH data points.
Should we take a regular sample or just a,
No, just, yeah.
do we have leftover filters? or
We do
don't?
have, do you want me to take a regular?
Might as well, I think. Before we stow it, actually, could we maybe take a regular super? It's only 10 minutes and have, how many leftover filters do we have?
We have, so we're on super, this would be super 11, so.
Yeah, I think we should just do it. Otherwise, it's
Okay.
no reason to come up with empty filters.
Okay.
All right.
Is it fine if we have no control then for this dive?
It's fine for me. Do you want a RNA later control?
No.
Okay.
Okay.
But even so, we would still have them. We'd still have one, right? There's
So
14.
there's 14, so, yeah, you're right, we would have one.
Okay. Yeah, so I think that's fine.
Okay. So, let me just prepare the purge for
No, this is not near orifice.
No, near Riftia.
Yep.
Yeah, that's
Wait.
diffuse.
Did we move over to the Riftia and I missed it?
No, we haven't.
Oh, okay. Are we standing by on instructions?
When we're doing another super.
Cool.
Yeah, do we wanna move closer to the Riftia?
No, I think having it paired is okay.
Okay.
Okay, good.
I'll say that the Riftia are, how far away is that, a meter?
Lasers, please. Though I've lost track of the worms now.
You're on the right.
Up
behind the
here or
behind the
there?
swat lobster.
No, they're further than that. That's why it was so blue and green.
There they are.
Yeah.
So, over a meter.
About a meter.
Yeah, maybe. Yeah, about a meter.
Purge complete. 11. Starting now.
You got headphones,
Your
right?
timestamp.
Ready for my timestamp.
You can hear everything twice.
Right, time is 11:09:40,
Okay.
and the latitude is 2.52,
Yep.
7362. Longitude is -94,
Yep.
0.075, 531. Okay.
Part of me wonders, Jacob, if this
Mhm.
was a situation like the EPR where when they took the crowbar to lift the rock up, they just found all the Riftia.
Oh.
You want to take that mic? People want to hear you.
Yeah, sure. Hi, this is Rachel speaking. Yeah, I was just mentioning to Jacob, so in the previous cruise that the Falcor 2 took to the East Pacific Rise, a couple of members of our lab who study animal microbe symbiosis, and particularly in the Riftia, one of the leading objectives for the cruise was to determine whether or not that if the animal biosphere extends down beneath the seafloor underneath these pillow basalts. And lo and behold, they used effectively a crowbar to lift up some of these rocks. And they flipped one over and they found that it was actually full adult Riftia tube worms that were present there, basically confirming their hypothesis. And so the fact that the very first Riftia that we are finding during this cruise are really wedged deeply into this diffuse flow fissure, you know, begs the question, how many more are underneath these rocks?
Thanks, Rachel. Maybe we should start carrying a crowbar.
What is this
I think that keeps locking out the autofocus. Or locking out the manual focus, rather. You know how to fix it? Thanks. Hopefully it stays put. Yep. Thank you. On the other hand, if this is kind of a new ecological regime taking root, it might be wise not to dig them all up before they get a chance. It would be cool to flip some rocks, though. I wonder if Stuart would be in support of that.
Flipping rocks?
In general, yeah.
What's one
See
rock?
what's there. I mean, they're all about the biodiversity index, right? And so, if it turns out that a lot of the biodiversity is out of sight, it could be useful for them to know.
Yeah. You can definitely see the diffuse flow right
Mhm.
here.
See if we can't find any others. Why stop there? Okay, I probably picked the wrong spot, but. There's a little kitty cat. Pushing down. Very nice little mussel cluster. Smallish ones.
Oh yeah, they blend right in.
Do either of you, Rachel or Roxanne, have any idea why the mussels here are so much blacker than at the other sites?
They're just bigger, excuse me, and older.
Roxanne was saying that the darkening of that outer coating on the mussels probably just comes with age.
Biofilms and exposure to the sulfur,
Mhm.
all of those things.
I personally am not familiar with any papers discussing serpulids as a successional stage on vent sites, but to be fair, I'm not personally familiar with very many papers about vent succession in general, just a few basic ones, so that's a possibility. I'll ask around. about the strapulids and their role in succession. Alvanellid worms are another type of polychaete worm that we often see in areas near hot venting, and we've seen plenty of them on this cruise. They're the ones with the sort of star-shaped red mouth parts. Lasers off, please. Do you think all this particulate's coming off of... Yeah, we're not near chimney 1. Where do you think this is coming from?
That was near Chimlet 1.
You think all this flux coming from the chimlet? Or is it just downwelling stuff?
Or the diffuse flow.
Hmm?
Right. Sample complete. You can stow the super wand. Do we want to get a pH rating right over the Riftia, or are you fine?
I don't think, I mean, one data point doesn't really help me in particular, but if you guys want to, I think he would want to know that you should do it.
Okay, yeah, let's move the pH probe over as close to the Riftia as we can.
Coming wide. And
'cause, yeah, we'll also get a temperature.
centering up. Yeah, I think I can imagine Pete liking that. Hugh. Okay, so our new target is...
Done with this one?
we can stow the super wand, and we're going to move the pH probe over here, which might require us to pick up.
Yeah, that's it. My little manipulator's about 2 meters too short
Yep,
for that.
yep. And we can lasers on whenever you have a free hand. So. Sorry, there you are. You let me know if you want the camera somewhere else.
I'll get it.
Okay, thanks. You're still in manual focus.
More important. Yeah. Working codes. Circles.
May I pan?
Yeah.
I think I need to. Okay, our target is right there.
Center of frame.
Center, yep. I'm really hoping some others will pop out of the woodwork here.
If we come around a little bit more, we might be able to see into the crack better.
I mean, don't go out of your way. Whatever, I'd say minimum effort to get the logger on
That hole.
the target. Yep. There certainly aren't enough of them to sample or anything.
How's this? This better?
I would change your heading if you're gonna come that far over, so you go for reaches. Oh, that's fine. I can push that. What is the camera? Direct out.
There we go. You think we can spare 5 minutes of log time?
Which Riftia, the one on the right or the left?
Maybe the one on the right, because it's a little bit more.
I just think you'll be able to get this probe more in the crack.
Yeah.
Yeah.
I don't know that.
Oh, I see another little. I see another little plume coming out on the
Oh,
left.
there's
If
a lot
there's,
of
yeah,
them.
if you move the rock, I can't be right.
Yeah.
Yeah. Once they get it planted, we'll look at it.
Now I know where you wanna go.
Yeah, just right down there. Oh, there's that one, and there's another one way, way over off the frame to left
All
field.
through the crack.
Yeah.
Yep.
Okay, let me just turn the heading a bit up.
That's awesome. I think we're counting like 6 at this point.
Yeah, I agree. Wow, what a fun thing to dive on.
I think you can just set it right down there on the rock in the crack. Okay, let's log till 11:36 UTC. Practically been there. Okay, we can go to 11:38. The 5 minutes really on site.
If it's more interesting to you, Roxy,
Yep.
I can forego the super samples at the chinlet if we want to try to flip this rock over to see if there are more Riftia.
I don't know how likely it is that we can flip this rock over.
One of these cobbles, like this one, see there's another one under there.
Oh yeah, look, there's
Yeah.
more.
So one of these cobbles might be flippable, but we'd have to defer to our pilots and kind of whether we think it's worth it time-wise and for the, you know, maybe low double digits numbers of animals.
Did we put a marker here for Riftia?
We didn't stand by on the pilots, but no, we don't have a Riftia specific marker.
We
need that.
We should change, yeah.
Which is one that H is kind of popped up right now.
mostly a lot of unhappy Riftia, unless you plan on collecting them.
I don't know what it makes sense to collect them. Both numbers.
It would get us a count, I suppose. And I don't know whether that's valuable. Considering we haven't, you know, that's not a uniform survey method for us. We've been running transects, but we haven't been flipping rocks.
Yeah, I'm not in favor of flipping a rock.
Okay.
I don't know what it would give us.
So you get your supers. You can see just a little bit of shear effect down there in the crack
Yeah.
on the left.
Yep.
We can see that they're there. That's a useful observation. It would be nice if we could collect them, but we needed, at least 10, and
That might be achieved if we flipped a rock, but they would be small.
And we've like counted 7 so far.
yeah, but they're very, I've got to look very hard at like all days, I think.
Right.
They're all in weird spots. But
Can we please drop marker Riftia 1? I know it's a marker pile, but this one's.
No, he had PH on Riftia instead of Riftia. I was guessing. He was guessing.
Oh.
Should have. The check's carried out.
Shall we take a count? 1, 2, 3.
Some behind them.
Yeah. We'll have to wait. I think there's 5. About 30 seconds to go.
We probably need to do like a survey of them for this area of Riftia.
Oh, yeah, that's right. Yeah. We should probably do a whole, like.
Some what all right, yeah.
Yeah.
The little
Figure out what
ones?
rocks we can really-
No, those are serpulid worms. So if we take a really close up look,
really close shot.
they are in fact tube worms, but they're in a different family. So like if you look at these guys, the mouth parts, which I think a lot of people think are the most interesting part of any worm, these ones are pretty much set up as suspension feeders. They have the feathery mouth parts. Those are all these little guys.
The Riftia.
No, these are serpulids, and the Riftia is that.
The pink one.
The pink one, yeah. Yeah. So there's a few of them. There's at least 5. Hmm. Yeah. When they make a living in a similar way, they're absorbing sulfide. They basically don't. They're Riftia, since they're obligated. They get all their energy from the symbiosis. That their gut is totally reduced. They don't have a regular digestive tract, as you think of it.
How was your coaching experiment?
I don't know. I wasn't in here at the time, but I heard it was a little bit cool. A little bit too cool. And they may try again later.
You wanna pull the page one?
All right. Yes. Thank you. Yeah, we can stow pH. Please. Actually, do you want to stow it, or do you want to try and super the chimlet?
No. Well, if it's fine to carry it, if we fly over to the pagoda.
I would stow it, because I feel like
All right.
it's going to be a
Stow
real big-
pH, please. Common sense prevails. Though we really don't have very far to go.
Jacob,
Yeah,
everybody can.
absolutely. Or would you be content leaving it in the quiver but keeping the jaws on it?
We won't be able to
Roxanne?
see where we're going.
Oh, okay. Okay.
But it's not hard to put it.
Great, great.
What you
Well, actually, while we have the porch out, could we open the bio boxes just to give them some fresh water?
Hmm.
I don't know if we need the sulfide, probably doesn't need it, but the mussels.
Let the mussels breathe.
Although, is that rock is for Nicole, or is that a?
Yeah, that's.
Okay, so then actually, yes, both of them, please.
It's too bad the aquafox doesn't quite fit inside the little ICL post. Convenient spot. I guess that would get in the way of sampling.
You want to open the buy boxes?
Mm-hmm.
Yes, please.
Open it all the way?
No, just a little bit. It's fine. I'm just trying to flush some fresh water in.
Maybe can we do a couple ups and downs to get some water motion? Yeah, those mussels must suck down the oxygen pretty fast in there.
Aquarium.
Yeah.
Treasure chest.
Yeah.
Do you think that's well, Roxanne?
Yeah, that's probably.
All right. Yep.
What you mean?
We got lucky with some of our super touchdowns. Benthic and
Right beside.
Yeah.
Riftia.
I think there's just more there than we realize, but unless you're sitting there looking
Yeah.
closely, you just don't see it.
All tucked into the cracks.
Okay.
Where are we going next?
We are going to.
Schmidt one.
So you're basically gonna come. Chimlet 1 is about 15.5 meters, 156.
Do you want to go to Chimlet 1 or Pagoda sampling spot?
The marker.
Pagoda sampling spot.
Okay, revise that to Pagoda sampling spot.
They're basically the same.
Yeah, they're meters away. If that.
It doesn't.
Yep.
I'm just trying to be extra nice because you gave me so much grief. You remember what you told me yesterday, Ben? No. You told me that if I was a nice guy, if.
Light. It was light.
Did I?
No,
No, I didn't.
light.
He said that we go to the sampling spot. Oh my gosh. Yeah, I can hear that. There we go.
He is a nice guy.
If.
Most of the time.
He said if. I didn't have his camera brightness adjusted properly.
Yeah. It was light. It was a position.
Here we go. He's in a.
Yeah. Farmers.
There you go.
We're gonna bring more rock off that one.
I don't have any sulfide sampling
We already collected.
marked there.
We already collected sulfides from there.
Yep.
No more rocks. Paper.
Okay, must be getting there. See.
Down. Have a wee look.
How we look.
Welcome to anyone who has just joined. We are still live in the final hour of bottom time on our final dive on the Natividad Vent Field, Western Galapagos Spreading Center.
You see it?
We're going to do one more series of super samples
I'd come down.
on a very cool looking chimlet, a small chimney.
Back up a little bit.
Then we're going to bring the vehicle home and we'll stream the ascent and see what we can see in the midwater. You're in the control room with Haley, Rachel, Jacob, and Chief Scientist Roxanne Beinert.
Ready?
Pilots extraordinaire Ben and Alex, and Media MT Josh. Hmm. Pilots ordinaire?
I don't know if that's it, but if you turn around, that might be.
If it is it, it should be clear from the other side.
Yeah.
I don't have a heading down for that marker. I kind of think that's it. Yeah. I think we took a few bites out of the flanges too.
Okay, coming down.
Do you want to look at it from the other perspective?
I think that would be useful.
So it's
Thanks.
around 90 degrees.
Yeah.
to start with.
Degrees to start.
Are there any flanges left?
I don't know if that's.
Oh yeah, I'm not sure we're looking at the right chimlet.
So that, yeah, that one had a very distinct
It had
structure.
3 distinct flanges.
Might have to look up visions.
Go ahead.
Can we gain a little bit of altitude? It's fine, look around.
So I think it might. So go further towards the marker.
Yeah.
Our heading was more north of this place.
Yeah, this is chimney 3. We need to go
You go to northwest.
northeast. Where it says
Oh.
Pagoda sampling spot, Chimlet 1.
Land. How far?
Like 10 meters.
Yeah, I'll go north. The grid's at 1 meter spacing right now.
There we are. And you're sit. See it?
Got it?
Yep, that's it.
Oh, and
Yeah.
assume Jill probably took the IGT from this orifice, because this is.
Well, it also looks like a nice healthy sample was taken here.
We just.
Oh, I see what you're
When
saying.
there's an orifice there.
Yeah, but I think her IGTs came from the other orifice.
Okay.
Down below.
The bottom one?
Yeah, the bottom one.
Okay, yeah, so that's
That's a
where I
good
would like to
spot.
super.
You know what? How high up?
Let's do about a meter, just to be safe.
So, let's
Let's do the. Want to do the porch first, so that way we have that out of the way then.
watching. Probably have to back up. I've got a stove chimney underneath the porch. Yeah, it's super right.
Yes, super.
Well, I'm gonna get the pH probe first.
Yeah.
'Cause you have to be sure you have.
Some soup.
I'm just gonna leave that for now until I grab that.
Sure.
Now, one of the PhD students aboard, Philip, has been taking biology transects. He actually got 2 done on this dive, and I think he'll be letting the AI loose on them for reasons that are already obvious to you all. But you know, to get accurate counts out of those sorts of analyses, you do need to train them on large manually annotated data sets. Without that, it's garbage in, garbage out, as they say.
Watching it. Yeah, rail into. Give me a little question.
Right now, Dr. Rachel is setting up for our final few super samples of the dive. For anybody who's been following since earlier parts in the dive, you might recognize some of these settings. We've retraced our steps a little bit because we were unsure that the super sampler would be capable of taking all the samples we wanted today, but it's performed really nicely, and so we've decided to come back to the same sites
Yeah, so
and take replicate samples with
probably like
in situ RNA preservation.
So like, right. Sorry, I thought you said a meter.
Yeah, sorry, just because that's a little bit lower. Yeah, just kind of trying to eye it by degree of diffuse flow.
That makes sense.
I could drive out to. Let's see close.
Ocular temperature.
Yeah, exactly. That's. Is it? Yeah, it's floating. That looks pretty good. Yeah.
Can you reach that with
You're about to find out.
Oh, those are flanges. So that's a particular mode of venting where the hot hydrothermal fluids come out sort of pointing downwards, and they deposit minerals above the flow. And so gradually the shelf gets longer and longer, and it forms something we call a flange. They look kind of biological. They look an awful lot like shelf corals, but they're actually geologic formations.
Okay. Alright, hold it.
Okay. We are on number 12.
They're just showing off.
Yeah, I think Rachel said earlier too, they look like turkey tail fungi on a tree trunk.
Alright, pumping beginning now for the purge.
I do like this better than just having a touch from here. I like having a table. Pressure on that. You can also just or like hammerhead console. I guess you can set that on the screen, but it scratches it.
Special lanes. It's still memories, lanes. Memory has been out for a while.
Hammerhead's been out for wait, oh my goodness. Almost exactly 10 years ago. Yeah. But it's changed a lot.
Yeah, I'm sure.
It has a back button now. It has a back button now. A back button.
Backlit.
No, a back button on the screen. And you go to the page, you get back and it goes back.
Oh, okay.
That was a brutal battle. South 13. Not because it was hard.
There's that.
They just didn't think it'd be useful.
Yeah, everybody likes it.
It was best upgrade ever.
Does it just pages back? It doesn't like go to your previous position.
No, it's just pages back.
Huh.
That's it.
So it's too hard.
Revolutionary.
To pick here.
So good.
Oh, I'm gonna go back here.
Well, here you have all your problems, like, cause you know how with hammerhead, so you can get multiple screens deep
Yeah.
into a menu, and instead of starting all the way back over to find.
Okay, purge complete.
Okay.
Moving on to sample 700 seconds beginning now.
Information. Okay. That one is 11:23:43, and then latitude is 2.5, 273.72,
Case is not.
Long.
negative 94,
Yep.
0.075,
Yep.
52.
2.075.
Yep.
Thank you. Hydrate, hydrate.
So, basically,
Hydrate.
is the joints in the regulator will roughly track the joints up here 1-to-1. If your scale, the 100%, so when you operate, you'll see the scale. You can toggle slow scale or high speed scale. This should usually be about 100%. this has less range of motion than the apples. So what we're jumping is going to be forming one.
Yeah.
Which is why you see us freeze it. But freeze it. That's pretty much it. Jaws are here. This rotates right here. You have the jaw bands to see, so you don't put your jaws here. This is the open. On the switch. That's it.
Mind if I look around a little?
Go for it.
Yeah, do it. What's it?
Find more Riftia.
So this is actually a trick close up. It's what it's called.
Aye.
The red button. Let's go outside. So don't do that. Okay. So they actually, what? Don't hit on twice. No, this isn't going to be an issue. This is a lot. This red button is. Okay. So none of the joints except for the shoulder actuator are locked. So if you disable hydraulics, like for example, on for them right now, it wouldn't really do the punch. It'll like the pitch will probably swing a little bit, and the elbow will come in.
Yeah, the shimmering
Did it on that.
is due to this optical phenomenon that we call the shear effect. But the shimmering specifically on the bottom side of the flange is really strong because there's a really strong change in density. All that hot water is pooling basically like an upside down puddle on the bottom of the flange, and it becomes very mirror-like.
Oh, no.
I've heard people aboard call it the mirror. You'll also hear us talking about shear, meaning shear effect.
Yeah. But I have this fun.
As distinct from fluid shear for you nerds out there.
This is what it looks like. Super sample. I think that's super. That's not purple. I didn't know it was right. I started to try to get the handle out of the box, and then I was like, wait a minute. That way you don't have to like thread it down into like two manuals. Yes, we did the same. And that we broke that focus. So we I mean, that was perfectly fine, but it wasn't at the angle that it is. What problem is Joe's really good at running it before. It's like he's not done for a whole
No, I think he's the one who booked the seat speed on the
Well, I know, but what I'm saying is, like running it, that is no problem. So, how is it? I mean, I think it'd be fine if it worked. I don't mind it.
I know I did.
There's definitely
There is a tiny larvation drifting up into the shimmering. Now you can't see it's that thing. It's probably an oikopleuron, about 2 centimeters.
What was it?
It was a larvation.
A larvation?
Yeah, like a larva.
Larva?
CEAN. At the moment, we are at 2,408 meters below sea level.
Is a larvation a larval form of an animal? Or
No.
is it just called larvation?
It's called that. That's actually a good question. The answer is kind of yes on both counts. A larvation is kind of like a tunicate that has evolved never to leave its larval stage.
Huh.
So they're pedomorphic, so to speak. They live their entire lives as the tadpole that a normal tunicate starts out as. They just never metamorphose.
They never grow up. They're the Peter Pans.
Yeah, Peter Pan's in the midwater. Well, the interesting thing is they show up everywhere. You find them on vents, you find them in shallow water, you can catch them off of piers, and you find giant ones in the midwater that spin these mucus webs that are over a meter across. There's a shiny polychaete back there, but the shimmer's keeping us from it. Try this one more time. Hey. Ah. They don't issue you calculator watches?
I wish. I don't know how much the green plan is.
Okay. Yeah, exactly. So larvaceans and their relatives tunicates, and also their relatives salps, are really interesting because out of all the invertebrates we see here in the ocean, they're actually some of the most closely related to us. The quote-unquote normal ones that aren't larvaceans, that is, tunicates and salps, they start out as sort of a little tadpole larva with a notochord, just like we have, kind of the beginnings of the central nervous system. And then in the case of a tunicate, which would live on the bottom, they find a nice little spot on a rock, they attach these palps on their head to the rock, and then they digest their own notochord and turn into an invertebrate. And those are the normal ones. Larvaceans never settle, they just keep drifting. And salps, I don't know exactly what prompts them to metamorphose, but they also digest their own notochord and metamorphose into a filter-feeding invertebrate.
Wait, they're not chordates, are they?
They are urochordates. So they're kind of the last split
Okay.
before the chordates. Not to be confused with hemichordates.
No, I know what hemichordates are.
Yeah. Whether you classify them as chordates kind of depends on who you talk to. It's like a lumber splitter kind of question. A very high level lumber splitter question.
3-domain, 2-domain, life equivalent.
In the animal world. I hope I'm not offending anyone right now. If you can get past the mucus and you look up giant larvation Bathocordius online, you'll see some really beautiful videos. They are stunning animals, and yes, it's pretty much all made of mucus. With any luck, we'll see some on the way up today. Ooh, no, those are all red worms. There's some tiny little Paralvanella.
Yeah, those are very little.
And tiny little mussels.
Tiny mussels.
And tiny crab.
Not as tiny crab.
See that berate?
Yeah.
Oh, he is tiny.
Oh, that's a little tiny guy.
Yeah,
And then tiny alvanella.
tiny.
It's like the nursery. The goat nursery.
Yeah.
And there's the anemone, just straight chilling.
Worms, crab.
That's pretty cute.
Remind me, are we taking a RNA and regular pair?
No,
Okay.
we have one more after this, and we'll do the mid buoyant, and then we'll be finished.
Cool. I'll check out that muscle moving on its foot. I think it just retracted from the hot water.
Okay. Complete. Switch over to SE port 2. 15 seconds. Pumping now.
Okay, Super 12 complete. Are we purging now?
No. We're
Oh.
already later.
Oh, good. Super 12 not complete.
Okay, complete. 5 13. Alright, we're ready to rise to the mid buoyant.
How high would you like the midpoint to be?
Go up about a meter. And about it there.
I can't really speak to the anemone's gender, but I believe they are separate sexed. A lot of anemones, however, can reproduce by fission, meaning that they just split in half and walk in opposite directions, and that's a form of asexual reproduction. So they have options.
Not a meter.
That's good.
Yeah, that's not
Oh, what is 5?
Okay, pump purging for under 20 seconds.
121340.
Okay.
And latitude is 2.52, 7245. Negative 94.075, 442.
Okay, thank you.
May I? You sure? You have stations holding? Yeah, just let me know. Hmm?
I didn't go see for Riftia.
Mm. Oh, do you want the side cam back? Okay. Okay. I think this pattern of parallel worms is interesting.
It really is.
Do you think they're just sitting in the little cracks on the pillow? Or do you think it's the current regime?
I mean, I feel like they're definitely aligning with the
Yeah.
striation on
Yeah.
the basalt.
Oh, there's the word. There's some real geology words. Striation.
Grad school, I'm in the geo department, I was like, I'm the resident bio person, and now I'm in the bio department, I'm the resident geologist. Don't know where I belong.
Yeah, so that was on the East Pacific Rise a few weeks ago. Uh-oh, okay, I'm coming wide. We fully expected to find large Riftia tube worms on this spreading center, in part because towed camera surveys and I think even ROV dives from the past had documented them. But we haven't found any big Riftia. We were very excited on this particular dive, possibly the last dive, which are the first and only 2 dives yet made on the Navidad field, which is where we are right now. At any rate, on this field, we were very excited to find just a handful of little baby Riftia, and so they may grow up to be giants if the sulfide flow and the temperature here hold and permit them to do so. But at the moment, they're just starting out. Whoop. Squidworm.
Squidworm. No. These are the ones that make me the most uncomfortable.
Yeah, it's like.
Well, then we'll get a nice zoom on this one.
These ones are, these little clear ones aren't as creepy to me as like the dark green ones.
Yeah.
Yeah, this one looks pretty small.
I was about to say, it's also smaller, so it's
Yeah.
less threatening.
Yeah. Like, we saw one yesterday that was like the size of like the front, the forearm of the net.
So these are relatives
Absolutely.
of the no manual focus. That's okay, it's swimming away. Those are relatives of the swimma bomber worms, though I don't know what their bioluminescence scene is. Lone mussel. Toothed Willis, that's right. Oh. Hmm.
It's been a long dive.
Oh, lost it.
It's like, that's enough. Not looking around.
So I can't grow it for long. It looks like it's.
It reads, but it's like hanging by a thread.
Physically broken.
Quite precarious.
Oh, I see.
And we're unsure of its absolute accuracy.
It's probably accurate. It seems reasonable. It seems like it, yeah.
It's probably close. It's a, it's an analog probe. So if there were water leaking into the leads, it would probably just result in like an offset one direction or the other, a small offset.
Lots of worms. Not the worms we are looking for.
But I like your pizzazz.
No, they're like little pom-poms.
Thank you. What is that? Did we leave that there?
Oh, zip tie. Oh, that's from Jill's IGT yesterday, I think.
What in the world is
And
that?
There's the one rock that
Is that a sponge?
Is it a sponge?
What is the? What?
It's unnatural, that's what that is.
It looks, I mean, to my subtitly trained eye, it looks like a yellow puffball sponge.
Yeah, indeed, subtitle.
Hmm.
Subtitle.
Yeah. Well, there are tides down here. They're just a little different. Maybe observe yellow sponge question mark. That would be a Demo sponge, which I haven't seen many of. It's pretty big too,
It's a monster.
20 by 30 centimeters. In my first years as a baby marine biologist at the local aquarium, working as an assistant aquarist, the head aquarist's motto was that mucus makes the world go round. And that was probably the most valuable thing I learned from him. Everyone has mucus, and pretty much everyone uses it.
Complete.
Coming wide.
Speed port 2. 15 seconds. Pumping.
Yeah, these small white worms, for those just joining us, are serpulid tube worms. They are tube worms, but the large tube worms typically associated with hydrothermal vents belong to a different family. They're known as ciboglenid tube worms, and those are the things with the big red plumes like Riftia and the Jericho worm Tevnia.
And we can put the pH and super wands away, but don't turn off power yet.
All right, I'm off camera.
And we can begin our ascent.
pH versus.
I think I might just do, once we get everything stowed and up off bottom, I'll do just a quick control sample.
Hmm.
Sorry.
So much effort for me to do.
The temp probe really doesn't look compromised. Well, I mean,
It looks a little sad.
mechanically sure, but electronically it's probably fine.
Power sirens probe rescue. Only could relay data real time.
I think it can. It would just be an integration problem. You know, a little wet mate connector on top. Pretty sure can talk real-time serial. Yeah. All right, let's get out of here.
300?
Seven.
It's a good depth. Yeah, it's a good depth because that's when we should turn out.
Do you have a maximum ascent rate you want to keep for the first part while you're taking this control?
Good to go.
No, because we're not actually pumping anything through the filter other than the RNA later.
Understood. Thanks.
So it'll just be 15 seconds.
Cool.
Bridge.
But I will start the purge. Starting purge.
Bridge ROV.
Yeah, go ahead.
We are off bottom.
Off bottom, copy.
All right, for everyone tuned in online, thanks for joining us. That concludes our benthic activities at the Navidad vent field on the Western Galapagos Spreading Center. We now have almost 2.5 kilometers of ocean to traverse on our way back up to the ship. So we'll do our best to find you some gelatinous and other forms of zooplankton and micronectin, denizens of the habitat that makes up about 93+% of the habitable volume on our planet. That's the midwater.
Like yesterday, you got the
Can I tilt up a little?
temperature falling.
Yeah.
Sure.
And now you look at me and you're like, "Jeez."
Purging refers to clearing the tubes on the super sampler. We just don't want to get the samples mixed up by having water from one site sit in the tubes and then get pumped into a filter that's supposed to be loaded with water from another.
Is this yours?
The second time you lost it.
It's just
Is it yours?
sitting here.
Or maybe that one is Pete's.
Maybe this was Pete's. I'm like,
It's too small to tie in a knot. It's too small to attach to myself.
Right,
It's good as
purge
last.
complete,
Um,
sample 14.
is the SciCam port of center? I just want to get oriented based on that pencil cam.
15 seconds.
It's centered?
Like you mean our
Centered left or right? Yeah, how does it look to you?
Location.
Oh, there we go. You can brighten it up. Nice. All right, pretty darn centered. Yep, thank you.
oh yeah, we can disconnect it. Yeah, thank you, I appreciate it. Yeah, go ahead and disconnect it. Yeah, we're good, thank you.
Oh, that's cucumber. That looks like a small siphonophore. Does vert rate read out somewhere here?
Yep.
Where's that?
Next to RV, meters per minute.
I see, thanks.
Okay, you can shut off superpower.
Super's off.
Thank you. Timestamp on those.
Yep. That is.
Pete's not.
Time 12:33:50. The lat is 2.52, 7249. Longitude is -94.075451. Next course. There we go.
So it's been requested that once we get to about 500 meters depth, that we power on the super and just, I'll cycle it through some of its commands just to see how it's behaving.
That's not all you got. All you got. Oh, 500 meters. You should be able to do it once go.
So we won't be taking any samples, we'll just be testing it, seeing if it has the same issues.
There was no samples
No
You just have to
samples.
move it in that off.
Yeah.
Have we filled the super cassette?
Yeah, filled the carousel.
all 14.
All 14. We did
I
it.
imagine he'll show up eventually to
Now
fly.
it's time to find us.
I thought I saw one on the descent for this dive, but
Mhm.
it was very, a very chaotic time.
Yeah.
We probably did.
We
Oh,
stopped.
for midweather.
Yeah.
For midweather.
Yeah.
Trying to get to the bottom.
That's why I'm just kind of running. Trying to get this.
A lot of those shallow animals.
Water.
Yep.
Does that
Yep.
really interest me?
Yeah.
I mean, it does. That's like fishing. I think fishing's boring.
So basically any large targets we might want to fly on. Possible. For anyone joining us at the very last minute, you've caught our ascent from the Natividad vent field on the Western Galapagos Spreading Center. You're in the control room with the Dawn Watch, Haley, Rachel, and Jacob. Pilots Drew and Alex on the sticks. And it's going to take us well over an hour to reach the surface from here, so stay tuned and hopefully we'll see something interesting on the way.
And fight, don't forget fight.
Fight back there? He's hiding behind Drew.
Yes.
Yes. Fight is basically responsible for everything you see and hear. Otherwise, we wouldn't know how to work the computers.
It's Fight Appreciation Day.
Okay.
It's always Fight Appreciation Day. We'll pick you some plastic flowers from the lounge.
Flowers.
Yeah, that's part of the appreciation.
Rachel and I are actually postdocs in Pete's group, and yes, he definitely has the narration thing down.
We could do our best Pete impressions though.
Oh, we could come up with some really good Pete-isms.
Yeah. I would put my friend hat on. Say, over bend. We should set.
Oops, we'll keep going. Okay. Are you okay with my potentially calling out stop? If you
Yeah.
can't make it, that's fine, but that's
No, I just-
what I'm accustomed to for midwater hunting.
That's fine.
The ascents are relaxing for us too. Maybe not Jacob, but
High stakes for Jacob.
These scents are relaxing for everyone until I get too excited. Oh.
this was a long dive.
Mm-hmm.
Yeah.
I think it was our longest one yet.
Yeah. I think you're right, for sure. Had a lot of objectives that took some time.
Can we slow down to maybe like 22 to 25, please? That's
You can get that live also. I don't know the version in my head. It doesn't smell in here, it smells everywhere.
maybe we target 22 and a half.
It's the screen, it's protecting us. I heard.
No. Yeah, take. Oh, stop if you
I got
can.
a whiff back as soon as you opened the-
All right, down right.
Can't leave.
Stuck in here
A
now.
little bit, feel like-
Yep, I'm pointed way up.
There it is.
Close the water
There it
tank.
is. That's a worm. We don't need it. Okay, won't zoom.
The floater.
Yeah,
And also if you could not move the camera off center.
sure.
Yeah.
Would you like me to pan back to center, or you want me to leave it for now?
Yeah, you can wait a second.
Okay. Panning back to center.
Panning while somebody's flying
Yeah,
is
no problem.
evil. It's absolutely.
It is evil?
Mhm.
It's evil.
Why is
Oh,
it
nice.
Because if he
Turn off the.
pans left, it makes it seem as though the ROV is going that direction.
So that means-
And you start trying to counter
So that
that.
means-
You're constantly putting a command in to keep it in the center, but it's not staying still. So now you overcompensate and it just shoots off screen.
I see. I thought you were
For
referring
these
to worms.
purposes.
Nope.
Sure.
I knew what
Opening
you were
iris.
referring to, but I was like, nope, we're about to find out.
Just hear
anyway. And that makes sense though, I like that.
We can keep ascending. This isn't this is a floater worm, somewhat related to the squid worm we saw earlier. It belongs to a different subgroup of the acrocerid worms. Oh, this one looks like it might have eggs. Yeah.
As usual. What's their delta?
Depth.
73.
We're still hanging in though, right?
I don't know where I stopped it when you stopped.
Oh, okay. Why are
Yep.
we stopping?
So those orange things look like eggs there.
Oh,
Hmm.
like for, okay.
Okay.
Have we been stopping it every time?
Oh, this is our first stop.
We've not been on clock.
Can I come wide?
Okay.
Ready to go?
Yep. So if it works for you, I mean, you know what kind of schedule we're trying to keep, but, well, I don't need to tell you about ascent at all. If we were targeting like the low 20s.
Right now we're on the INS. You want to get CTV?
The low 20s?
Low 20s ascent rate. if you think we can sustain that and still get back in reasonable
yeah,
time.
we come up at 24.
Cool.
24, 25.
Earlier we were doing 30, so 24, 25 sounds
Well,
nice.
oh, the ROV. You're talking
Yeah.
about the ROV. No, so the winch goes in at a constant rate,
Uh-huh.
and so I have to speed up and slow down.
Sure.
So if you're wanting to look at a certain water column
Mm-hmm.
where all the goodies are at,
Mm-hmm.
we-
Whatever you were doing before was working great.
Yeah.
So I'm not going to micromanage. There's a cucumber. Is it all right if I just call out stop if, or feel free to stay if we see something that I like, or feel free to stop if you see something you like.
That's the black water.
And has the slurp been working today?
I don't know that we've tested it.
The other techniques are-
We've not had any slurp objectives this time, so we did not attempt to slurp.
I'd be happy to find out though.
Can you grab the slurp? I will get it ready when.
Yep.
Try to get you a view of it. See if I can point it out a little bit. Actually, if you use your T4 cam, it's probably going to be the easiest. I can porch out a touch. So you're not in so tight. No, that's not doing it. It's going to be the rail.
Yeah, my grandma used to like to say that you only really get to know someone by living with them, doing business with them, or playing cards with them. And a lot of cruises involve all three, so we get to know each other pretty well. I haven't played any cards yet either, but it happens.
You know how to learn how to play euchre. All the Michigan people on here. Or
Michigan
British.
people. No,
Well, no,
Michigan.
I'm just saying we have a lot.
Oh, we do.
Yeah.
Yeah, you're right.
Is that why that chimney ended up being named Michigan?
It looked like Michigan.
It looks like Michigan.
Yeah, yeah.
Like little
Gathered that.
mitten, little mitten.
We'll do one with respect. There you go. Got it.
We've got Carolinians in the room.
Yeah.
I'm north.
You're north.
Haley's south.
That's right. We got the Carolinas covered.
Okay, so now, let me know when you want me to bring that rail in. Let me see if it'll go now.
Yeah.
Oh, watch it, you got shoulder left. Your forearms on the end of it. Okay, shoulder left.
I'm like, there's no way it's gonna keep that. There we go. There's no shortage of that. I know, it's like on tap. It's on tap.
I wish. We're always running low. Alright, we are ready for potential
Sweet.
I know, we used to at one point, Clyde would be able to say if we can still do it, but they used to have one of the sonars on the boat that you could actually watch that migration happen.
Mm-hmm.
And so that one area.
An EK fisheries sonar?
Yeah,
Yep.
and you can slow down in that area, and that's where
Everything, yeah.
it's gotta be thick, and
Yep.
going through it real fast is gonna miss a lot, and so.
Yeah, the deep scattering layer. I don't know where the DSL is here. It's probably at the equator. It usually comes to the surface at night, so it's pretty shallow.
Well, it should be on its way down right now.
Hundreds of meters, yeah.
Hey, Mike, do we still have the?
EK80?
You definitely.
Do we have an
Yeah,
EK?
we have EK, it's full swing, but we haven't got anything on,
Okay,
because it interferes with the USBL as well.
gotcha.
But what can be useful is just a CTDO trace or CTD trace.
Oh, the O2 level, yeah.
Mm-hmm. Actually, for what I'm
Looking
interested.
for the minimum
Mm-hmm.
zones.
Yeah, which has some really interesting stuff living in it. But at the depth of the minimum zone, it tends to be kind of sparse. Drew, are you ready for me to ask the most Steve Haddock question ever? Do you know what's up with the stripes on this monitor?
Not my monitor.
Yeah, but I knew you'd get the reference.
Yeah.
We can't figure it out what's causing that one. If it's the monitors themselves or if it's actually the video, but I haven't. Sometimes you see in the video if we have the floods on and our shutter speed is off. But
it's nice and regular. It doesn't really interfere. You know, it's not like one of those splotches on the dome that looks like an animal all the time.
But it should be the same signal that's going to this monitor.
And I don't see it for. No, it's there, it's still there. Yep. Different viewing angle.
over there as well. So I'll have to look at the recording.
Do you think it could be a beat frequency between the PWM drivers and the DSPL lights? I imagine there are.
If they're dimmable, yeah.
They're not true analog. I don't think they're.
You think it's
I don't know if they're nominally dimmable. There's a safe.
So we're running a different hertz rate than what we used to run.
That's always a good guess.
I mean, if it were, you should be able to shift. Well, the frequency, the PWM frequency would be consistent regardless
Of your brightness. Yeah.
So I don't think you can change that as a user. That's more of an engineering level, some circuit design sort of thing. You might be able to shift the shutter speed on the camera that would come in and out of sync with.
I don't know it. To be clear, I don't know anything about these lights. No.
Yeah, and that's
That would at least move them.
Yeah,
Right?
Well,
That would-
you would change your frames per second, not the
right.
depth.
Not the integration time.
Not the length of the shutter being open.
Sinker.
another.
Cuke.
Is that the Halothurian?
It's a Holothurian's a fancy word for cuke, and it's a Pelagothuria, I think, is the species name. Ooh, who is that? May I manual focus? Thanks. That's interesting. Ideas, anyone? Let me know when I can zoom. Standing by. Huh. Is it really?
Yeah, just a tiny one.
Now that you mentioned it, I can't unsee it.
A little planetary nebula.
Yeah. Just like you said, pixels.
Yep.
Thanks, Jake.
Live to serve.
Yeah, it's stuff like that.
I think it's an unknown.
You're good to zoom in if
Huh.
you want.
Cool. Yeah, it's a great illustration of just how delicate some of these creatures are.
I don't see the swimming bell on that.
It
so it
kind
might
of-
have just been one that was already broken.
Yeah, it might have been a chunk of tail. It looks a lot like an Apolemia species to me, which are these ones that get really, really long, like tens of meters long, if not longer. And I don't know whether it was just a broken off piece or like a tiny baby, or both.
Are Apolemia light sensitive?
Are any of them light sensitive? Almost most of them are at this depth.
Well, that's specifically asking about Apolemia, but
Oh.
I'd say most of them are.
I think. I'm trying to think of the last time I really zoomed on Apolemia nectosome, like the head end. I think so, but I can't give you a solid answer. Yeah, do you actually, not to harp on this, but just because I'm curious and we're moving through blue water, do you have a frame rate adjustment or is that fixed as part of the codec?
We have a frame rate on the camera, but
Mhm.
the recording is locked in at 30.
Of course.
So it can come up Alex and say we can mess up the recording if we're not
Yep.
on it, but it seems to be working, so I don't understand how that messes up the recording.
What,
Well,
when you say you're adjusting the shutter speed.
Right.
But, so that's how long the shutter's open for,
Right.
for your frame rate. So you can have a shorter shutter speed, like the 1/20th of a second, but you're still only taking a frame 30 times a second.
Right.
So as long as you don't change that, you can lengthen that shutter speed up until a point, you know.
Right, but he was saying we had to stay at a multiple of 30.
Right. Or,
So the only ones that said 30, 60, 90.
yeah.
Yes.
I don't know, like, I mean, because if we go to 30, it actually probably would help us, because we could get more light in, but you'll get more streaking as well.
You said it tend to stay at 60.
We tend to stay at 60. That's the default for the camera. We've taken it up to 90 before.
Yeah, in theory, if that was an interference pattern between the lights and the frame rate, then if you doubled or tripled it, I think you should double or triple the number of stripes, right?
Yeah, I think if this is a monitor thing.
Lagothuria.
Well, but that's common between two different monitors, so unless it's something.
They went through in between the last.
You're just
two,
listening into the pilots and I discussing the joys of
like, adjusted
video engineering.
And
There's
this
some
one
very
started
subtle
dripping.
stripes on our screens that are only really visible
So,
in open blue water.
Yeah.
they had adjusted them all once before, except for ours, which I told them not to touch, and these things did, and now that they've messed with it now. I don't know what I probably need to get there and just revert to default and see what we got.
Yeah.
We can play with it more on deck, the only problem is things are getting a lot of power as you go with the blue and everything.
Yeah.
It's almost like we'd have to replay the dive video.
Yeah. I'm very much I can, but it's like, just make them all the same.
It's so hard.
Even if they're wrong, just make them all the same.
I'm so used to flying like facing this way with it that but that's not the right lighting. I have to go by this one.
There's a Gigantocyprid ostracod.
You want to have a look at another one?
Sure, we looked at one yesterday. They're always fun. They're cute. I have a video that a friend sent me from Boston of their cat chasing it around the computer screen.
That is awesome.
Cat TV.
Yeah. Yeah. This is a Gigantocypris ostracod, or a giant ostracod. It might not look that giant, it's smaller than a golf ball, but compared to most of its relatives, it's huge. The ostracods are a group of crustaceans that are sometimes known in plain English as seed shrimp. They're not really shrimp, because nothing is simple, but most of this guy's relatives are about the size of a sesame seed. This one's an exception. Squirrely one. And they're a great example of deep sea compound eyes evolved to take in just about as much light as possible. Yeah, we can keep cruising. Mm-hmm. If you feel like adding a little bit of comparative morphology to your morning or evening or whatever time of day it is, you can compare those to the barrel eye fish, which of course is completely unrelated, but its eyes, despite not being compound, look remarkably similar. They're large and they're upward facing through a transparent canopy. The thinking is that those kinds of eyes can pick up the silhouettes of potential predators above. I bet we'll be able to tell the deep scattering layer when we get into it. We're still well clear of it.
Look at the oxygen.
Do we have it?
Yeah.
No.
Oh wait, no.
I think the Andera's out.
I forgot. Yeah. Right.
But temperature, and if we had a trace of temperature and
Definitely have temperature,
salinity, that would be great. Do you have like a strip chart of them? I feel like I saw one over there by the mapping computer the other day.
That would be a light.
It's a fight question. Do we have a CTD plot by chance? Only if it's easy.
From the RV.
Yeah, from the RV. Yeah, it was through Grafana earlier. My bet is we're going to hit it somewhere between 600 and 400 meters. We're stopping there anyways.
Oh, we are?
Yes, don't we have some kind of stop at 500 meters?
Well, we're not stopping, we're just gonna turn on the super and I'm gonna cycle through commands.
Understood, okay. I heard Alex ask for auto depth to be set to 500 earlier.
Yep.
Yeah.
Well, if you want us at 500, we need to set that 560.
I don't think it's how
It doesn't need to
critical
be
is the actual
precise.
depth.
It
Oh,
doesn't,
okay.
yeah, it just
Roger.
around that depth.
Okay.
I'm just wanting to see if we're getting the same comms error as we're getting close to the surface.
Well, now that you got that squeeze, it should be fine.
It won't depressurize as we go up?
Typically, no. Typically, once you get that good squeeze on the connector, it stays, but it has happened before to where, just like our parent depth to that doesn't like to work below 2,000 meters.
At less than 2,000 meters? Or deeper?
They like to work on the surface, but it was fine on the surface, and then
Do you think that's a gas pocket in the wet mate connector? Is that what's going on?
On theirs,
Yeah.
oh, just thinking, yeah.
Yeah.
Yeah. So once you get that squeezed out.
So it goes into solution. We've been seeing pretty nice weather, all told. I don't know what else to say about it. It was ideal last night. It was probably in the 70s Fahrenheit.
the seas were a little rough a couple of weeks ago.
Yeah, we did get to some big-ish groundswell over the past couple of days. Fortunately, the Falcor 2 is a very stable ship.
569.
Yeah, that one.
That's
Yeah. Never to be slick enough.
a nice setup.
A little bit of rain.
Sprinkled. It's like you don't have to spit out.
Better that than getting spit out by the birds.
Yeah, that's how I felt too.
I think it's another giganto. Yep. Yep, yep. Same.
Except not flailing around like
Yeah.
He's holding still a little bit for us. Thanks, guy.
the last guy was not
Uncooperative. There he
Oh,
goes.
woke up. Yeah.
As soon as you said that, I was like, no, but you're not gonna finish that for me right now.
So their shallow water relatives, known as sea fireflies, actually do little pulse patterns of bioluminescence they leave in the water for mating, just like fireflies have flash patterns.
I love that.
There's a great video of that on the Oakley Labs website out of UC Santa Barbara. That ostracod is full steam ahead. I'm almost certain I asked you this already, but I forget the answer. When you were diving with Dougal or any other time, any of the classic expeditions, have you ever carried detrital samplers?
No, we got them during the refit of going from Falcor Classic to Falcor 2. So we have them.
Oh, fantastic.
They've just never been used.
Okay. Pete mentioned in a meeting that you might have them. I was kind of flabbergasted.
Yeah.
That's very cool.
Yeah, we haven't used them yet. They're
Where-
just big, so they
Yes.
take a lot of room.
How many did you get?
No, I think we have like 8 of them.
That's not too
But-
shabby at all.
But I mean, I don't know how we would store 8 of them.
I'm not suggesting that we that we mount them. I know they take up a ton of payload
This is
space,
not the tenacious team for
but it's really cool to know that that's available.
expedition.
Are they all hydraulically actuated?
Not yet.
I imagine we can make one.
There might be a new thing.
Do you need to add a valve pack?
Hmm?
Do you need to add a valve pack to run them?
Like, we could mount them on the front porch. We have no file boxes and all
Of
that.
course.
We could
Yeah.
easily put 6 of them on there.
Mm-hmm.
Or load up. Looks like load up.
Take a look. So for those listening, detrital samplers or D-samplers are these sort of
Load up.
jars with hydraulically actuated sliding doors that allow us to collect some of these really fragile midwater animals without actually touching them.
They look like the things that they used to use at the tank.
Yeah. Never thought of that.
Was that
Hmm?
Oh, I thought that might have been a
It is.
Tina
That's
Ford.
all. It is. I'm pretty sure Tina Ford down there.
I thought that was a seat cover.
Oh, you're right. It's
In the back.
yeah. J Rod knows it's a cuke. It's a
He knows
it's a Pelagothuria.
Yeah.
I was wishfully thinking that it was a lobate going down towards the right.
That's what I initially thought.
Yeah. That we're kind of on the lookout for that lobate though. If we get another one, that would be nice. The one we got the other day was great.
How'd the one that came up?
I mean, it was messed up because we took it on a whole dive, right? But it had DNA.
We dragged it around.
We caught it at like 500 meters and then took it to 2,500. Towed it around for yeah. It was still kicking, but it
But
a little worse for
Did
wear.
it get torn up?
Yes, but not. Well, let me put it this way: torn up, probably not. Just disintegrated over that course of time. Yes, but no fault of the operator.
I can't pull the Grafana picture because there are too many data points.
No,
It's
no.
too big.
That's too big because it's so long, so I can't pull the
Oh,
first
yes.
so it's 4,000 or 6,000.
Sure.
It's total maximum I can pull.
Yeah, no big deal. Thanks for trying.
There was a limit, I know that, but yeah.
Mm-hmm.
I've got the same problem at home with the weather station.
So it won't just take the like and it won't just take the 600 most recent points or whatever.
No, I think it's
Huh.
it's truncated here. It's truncated to 9,021 points.
Uh huh.
So that's a set in the green or they can just set up and say what's maximum stuff that we decide to look at.
Oh right, and that'll only give us our bottom data, right? Yeah.
But it's
Okay.
just get the last bit to visualize.
Well, thanks for looking.
This current just changed 180 degrees.
Huh.
Maybe 500 meters, maybe.
It's the Ekman spiral at work.
Yeah, well, I mean, I fixed this way at first, because the current was
Mm-hmm.
going that way. And going, and then it changed going this way. Two of them. One big, one small.
I forget the equation for the pitch of the Ekman spiral, but my recollection is that the faster the current is, the faster it turns. Does that sound right to you? I don't know if it's linear. So, because of the Coriolis effect, we're going to need to get a proper like physical oceanographer to explain this to you. But basically, as you go down in the water column, the heading of the current rotates uniformly, and it's named after this guy Ekman. And you know this, I'm sure, on a practical level. And there's have been decades of mathematical modeling done to figure out how tight that's. But like, if you think of it as like threads on a screw, like what's the pitch? How tight that spiral is. Oh, that's a scythe. Unless it's one of the ropes off of our lander that floated away. Let's check that out.
Yeah, I'm trying to get away from him a little bit.
Yeah, so the nectosome, which means swimming body, or the quote-unquote head of the siphonophore, is on the right-hand side of the screen. And then the rest of it is all just one big mean eating machine. Those are gastrozoids. Zoid is kind of one of the small individual animal-like units that makes up the animal. Gastro means stomach. I don't know who this is, but I think we should get some good frame grabs. It looks like it probably has fluorescent tentacles. You can see kind of a faint greenish tint at the end of the stinging tentacles. Beautiful.
Too dark, or is that
Fine
fine?
by me. So if you annotate this, yeah, if you just annotate the siphonophore, that's great. If we get to the part that bends over, I wonder if we can get lasers on it. It's always tricky in the midwater.
Come on, lasers then.
Ready?
Oh yeah, we can get lasers right on this fishing section here. You think we can get the? Well, that's fine. We'll just go to a place where it bends over. Yep.
Right here.
Yep. So you might have noticed those stinging lures hanging down, and there's actually been some really interesting work done on how the shape of those lures compares to the shape of little midwater crustaceans, like amphipods, that the fish that siphonophores like this feed on might want to eat.
Good enough video with the lasers on it? Or
Yeah, that's good. That's good. We don't need a frame grab with the lasers. If we are really determined, we'll track that data down.
Kill the lasers.
Thanks. Yeah. Off we go. Thanks, J-Rod. How long have we been on this Galapagos expedition? We just passed the halfway point at 2 weeks. We're basically a month out to sea, and I think some week out of that in total is going to be spent underway transiting between sites. So you should have several more dives to look forward to, but our next mission immediately after we surface from this dive is to start tow-yoing and looking at new potential vent sites. So it's a little bit difficult to predict the exact date and time of our next dive. It'll depend on what chemical signatures we see in the water pointing to a vent we might be able to discover. Do you remember where you had the D-samplers made? I'm curious. You probably have a lot of vendors in your head.
They were probably made in Canada.
Mm.
Imagine
The
that.
same outfit that did the suction unit?
Probably not. I mean, I think we got our original designs from Bari.
Uh-huh.
So-
And they got their designs from Harvard Branch.
Yeah.
They're all the same.
Yeah, or maybe we got the Harvard Branch, like
Yeah. They're standard, which is nice. One of the few sampling tools I think that's really standardized.
I wish bioboxes would be a little bit more standardized.
Well, milk crates are standardized.
That is.
Oh, bio boxes. I was thinking of rock boxes. But that's a hydromedusa. We've seen a few of that species. I can't offer much more on it.
Really tiny one in the background.
Yeah, that's a colubanema, I'm pretty sure, in the background. That's a different kind of hydromedusa. And when we see them, they tend to make a break for it like that, and then they drop their tentacles. Actually, yeah, that's a colubanema, so it has mostly dropped its tentacles. Right? Isn't that who that is? In fact, the one in the foreground may be one that's already dropped its tentacles. It might be the same species. Before somebody asks, yes, they probably grow them back, though it's never been conclusively demonstrated because we can't keep them in captivity yet.
What's the major limitations on that? Is it the pressure or the temperature?
It's a good question. It's difficult to know. So J-Rod just asked, I think you could probably pick him up there, but J-Rod just asked what the major limitations are in our ability to keep some of these animals in captivity. It really varies depending on the animal. Some of them, we figured out, the main limitation that prevents us from keeping them in normal surface seawater is that they can only handle so much oxygen. And if you give them surface levels of oxygen, all the free radicals that form just eat their tissue. That's a pretty easily solvable problem compared to something like temperature, because all you have to do is blow a bunch of nitrogen on top of the tank and strip the oxygen out of the water. So at the Monterey Bay Aquarium and their deep sea exhibit, for instance, those are all just low oxygen exhibits. Some of them do appear to need pressure, irrespective of oxygen. And pretty much all of them are sensitive to temperature. If you overheat them, they're not happy.
It's crazy how tolerant crabs are.
Yes, I've noticed that. And it depends on the species. But another crazy thing is, how oddly specific it can be. But within crustaceans, they tend to be pretty hardy. When I was in grad school, we were picking, I think, benthic ctenophores and sponges off the bottom. We accidentally ended up with this little decorator crab, and it just sat in a dish in a cold room for like a year, crawling around,
No way.
eating stuff, just chilling, 4 degrees Celsius.
Awesome.
Remember the ants? Complete different story. The ants that were in the green ants gym.
No.
Oh yeah.
So there was this guy who does originally recipes in Darwin,
Uh-huh.
northern Australia, and he does traditional recipes, and some of them contain ants.
Okay.
And when he experimented with the food he put them in the freezer at -30, but then they got enough. So when you defrosted them, they started breaking up. They had nation. So we had to go to -80 minimum in order to
Wow.
kill them.
Yeah, some insects are incredibly freeze tolerant. Most of them use these compounds in their body fluids. Trehalose is a really common one. It's a sugar, and basically what it allows them to do is that normally when water freezes, it crystallizes and expands a little bit, right? This is like why jars explode in your freezer. But with these compounds laced in between the water molecules, they can form what we call vitreous water, which is glass-like. So instead of the water molecules forming into a crystal lattice, they're still randomly oriented, but they're a solid, and they don't expand and the cells don't burst. And then when it melts, they wake up.
We've recovered worms from Siberian permafrost. It's been frozen for like 18,000 years.
Annelids or nematodes?
Nematodes.
And they just came back to life when
They
you
woke right up.
Yeah. A lot of these animals do absolutely require pressure. I'm actually working on a paper right now that indicates that one of the main reasons for this is because their cell membranes, the structures that are made out of lipids, made out of fats that basically wrap around each of their cells and keep them contained. What's that?
Fish.
Okay, thanks.
I'm interested in fish.
It's a shrimp.
No, the one.
Oh, okay.
That thing that's flashing at us
All right. Oh, yeah.
and swimming away.
It's gonna be hard to catch. Anyways, those cell membranes are really specialized for the pressure that the animal lives at, and some of them actually require pressure to hold the membrane and therefore to hold the cell together. When you release the pressure on the organism, even if you keep it cold, even if you keep it at low oxygen, the cells just kind of come apart. We did bring on this expedition a couple of... Oh, who's that?
I think that's one of those little octopus-like things. It's
Oh,
not an octopus, but it's like in between a squid and an octopus.
a cirrothalmid.
Looks
Yeah.
like a really adorable octopus.
Uh-huh. Well, then we're here for it. Oh, is that a cockeye squid?
No,
No.
definitely not.
No, you're right, yeah.
We saw these on the Octopus Odyssey cruise, and
Huh.
we actually sampled a couple of them.
Yeah, we...
They're really adorable.
We brought some high pressure recovery vessels on this cruise, just a pair of them, but they are so heavy that we have so far opted to keep them off of the vehicle and the lander. Also, the ones we brought are more for collecting things like rocks and microbes. As far as I know, nobody's ever really built a workable pressure retaining midwater sampler. That's really cute. You, anyone know what it's called?
I can't.
Aside from a cephalopod?
Great.
Pinky in the brain.
Yeah, Gran Rojo, which somebody mentioned, is a type of jelly. This is basically an octopus, or closely related to an octopus. But red coloration is very common in the deep sea, across all different kinds of animals.
No touchy touchy.
Maggie's been putting together a nice little field guide of frame grabs that we've seen.
Nice.
She can add to that. Yeah.
I think Sophia has a pretty good one too.
Oh, that's awesome.
I used to
Has she
do-
published it anywhere?
I don't know. You probably can ask her to. I used to before that position existed.
Mm-hmm.
It used to kind of be my job, and I would just have a folder in the cruise
Oh.
folder that was just J-Rod's favorite picks and clips.
Oh, that's awesome. Love to
Now
see
I
that.
don't have to stay up after shift anymore.
You did double duty? That's pilot and outreach. Yeah.
The MT, it was a mixture of the MTs and us doing different aspects of what all Sophia and Alex do on board,
Mm-hmm.
which was a growing thing. It just kept getting more and more and more, and ultimately
Sure.
neither one of our departments could
Yeah.
keep up with it and continue to do our jobs properly. Now we have Sophia and Alex, which is
Mhm.
very helpful. I remember those things liking the edges of the oxygen.
Minimum zone.
Yeah, like they stay in the minimum right before it gets high again, right? Or no, right before it goes minimum.
Okay.
They like that area.
It's down the bottom margin. Is that what you're saying?
Bottom
Of
top,
the
like,
OMZ.
like they
Mm-hmm.
run the area.
Mm-hmm. Cool.
If I remember what they were explaining about those on that last cruise.
Cool.
You know, that's one of those little crown looking jellies.
Maybe. What's that? Any hunch?
It looks like a
Kind of does, but it could also be a Solmissus on its side.
yeah, I'll try to get there real quick.
It is a Borrelia bisicola. Can we get a little video and slurp it? Yeah. So this is a ctenophore from the class Baroidae, the only ctenophores that don't have tentacles at any point in their life. It's got that classic deep sea red gut, which is probably useful for it because all of the ctenophores in the genus Boroi specialize in eating other ctenophores, and ctenophores light up. So if you are a ctenophore and you're transparent and you swallow another ctenophore and it lights up, your position has been given away. And that's why we think they have this red gut lining that masks light from things that they eat. One of the cool adaptations that make Boroi the terror of the ctenophore world is that not only do they have comb rows on their sides like most other ctenophores, but they actually have these things called macrocilia, which are kind of like specially adapted comb plates that are in their mouth. They're pointed inward and they act as teeth for tearing through other ctenophores. Yeah, we can do a quick slurp whenever you are ready. You might have also been able to see in the close-up all of those intricate channels inside the jelly or the mesoglia of the animal. And when these have been kept in the dark for a while and they're disturbed, they'll dump really, really bright bioluminescent chemicals into those canals, and they kind of radiate in a, it's difficult to describe, a network pattern. Yep, this is Boroi. The closest described species anyways is Borrelia bisicola. Oh, it's a chonker. It's bigger than I expected. Thank you. Phenomenal.
It's actually smaller than that last one over there.
Yeah, they're pretty hardy, so that particular species. Thanks.
That little guy right there.
Yeah, who's that? That looks like a, we call that a spider radiolarian.
Yeah, super tiny.
Yeah.
Yeah, good thing.
Was that smaller than the other one? It just went in sideways.
Yeah, the other one, like, it barely, like, it hit the bottom and was barely out of the chamber. That's why we wanted to keep the suction on when we went by it, which ultimately was our downfall.
I'm pretty sure there are some nice videos of Boroi of one species or another bioluminescing on the internet. It's a highly recommended watch. Look up Boroi bioluminescence. Oh, I wasn't talking about our watch. I was saying that watching, though thank you. We do have a solid watch. I meant that watching Boroi bioluminescence is a good way to spend your time. And I'm pretty sure you can find it on YouTube somewhere. Oh. A real squid.
Real squid.
For a change. Ooh, interesting one. Yeah. Mardi Gras squid one of your names? Yeah. No, I don't think it wants to be collected either. Yeah. No, we don't have them on our permit. We do. We have ctenophores and siphonophores on the permit. And Stuart.
What about a squid trying to look like a siphonophore?
That's a question for Stuart. Yeah. So all of those little yellow dots you see on the skin are chromatophores, which are basically little pockets of pigment surrounded by a ring of tiny muscles. And that's how the squid changes color and transparency. So if it spreads out one of those spots, it's like turning the pixel on. And then when the muscles are relaxed, the spot contracts into a little dot that's pretty much too small to see, and the squid can be almost completely transparent.
So you're saying it's stressed out right now? No. It's putting on
One of the awesome things about deep sea squids is you get to see their internal anatomy. That white thing at the top, I'm pretty sure, is the gonads. The translucent thing to the right of the red thing is one of the two gills. The brain, I think, is barely visible. It's kind of donut shaped and just to the right of the eyes. All right. Boom. All right.
I will get up shortly, yes. I will let you take over. So I should eat in a second. Yeah.
All right, J-Rod has cleared the squid from the danger zone. And off we go.
Let me finish I should probably, you're right.
It's that time already.
Yeah. Once is the question, yes. Just like deliberately not answering it. My bad. No, I appreciate it. I appreciate it. I should eat breakfast, you're right.
Yeah. Yep. Ooh. Oh, we got an ID, pretty authoritative ID on that octopus.
What would they call it?
Japetella or Bolitania. Yeah, Japetella sounds right. Japetella species. Thanks for the help from out there. There's a Solmissus.
I feel like Solmissus is the most widespread thing that's of a decent size.
Mm-hmm.
Not the small little,
Yeah.
I see them in every.
Have you seen any of the purple ones on this cruise?
Purple?
You ever seen a purple Solmissus?
Like dark purple?
No, like a bright, sort of violet, purple crayon purple. The bell.
I don't believe
And the endodermal layer. Those are common in the Monterey Bay, but I haven't seen any on this cruise.
I have to hear over super cycling. Do you want any food?
It's going to take us a while to get to 500. Good morning, Andrea.
Are you going to take a picture now? How do you want to stay?
Do I want coffee? If you're offering. Where am I at here? Does anybody else want me to have coffee is the question.
How much coffee have you had?
Anything.
Already like.
Oh, how much?
Yeah.
Only 2.
Oh, okay.
3 beans each. Thank you. That was a long time ago.
Wait, what do you want?
Anything.
I got a
Anything?
jelly.
Trust you, yeah.
Okay.
Thanks a bunch.
I
Oh, that's a.
Yeah, what is this?
Huh. Wait. Yeah, that's a Solmissus up above. This thing down below
Yeah, is this another Mardi Gras?
kind of looks like there might be some.
It's another Mardi Gras.
Yeah, a different one.
Yep.
Oh, gee. Okay, so this. It's a squid.
One day,
There's a siphonophore to the lower left too. But let's look at the squid.
one day Mardi Gras squid is going to become a turtle. That makes me so happy.
I think we're already there. That day is here. So 25 meters a minute, averaging 15.
Yeah, so those, yes, you'll need to, put those marinate over. Probably need to come back. Yep.
Rachel, we probably have a half hour before we hit 500 meters. Realistically, 20 minutes to a half hour.
That's definitely.
Okay.
Yep.
Then I'm gonna go get.
Go eat.
Okay.
Do you want to see that same bottle brush siphonophore that we've seen a couple of times on Ascent and Descent? Or?
Irena is cool, and Galaxy is cooler.
Okay.
Yeah. We've seen a lot of Irena.
Yeah, the bottle brush one.
I think we're talking about the same one.
Yeah,
It's
no.
kind of whitish.
Like the swimming bell and the stinging area are almost the same length. Yeah.
Yep.
That was very abundant.
Mhm. I'm guessing they'll line up with wherever. They kind of line up with wherever the fish are, but I don't think they're really fast migrators. They just wait for the fish to come through.
There's a squid.
Planktotuthus, then. Yep.
Oh, but
There's
that's
some of those the other day. Thanks.
what we just saw.
Mm-hmm. For some reason, the only one I can remember of the pointy squids is Chiroteuthis, and we haven't seen any on this cruise. Looks like it. Moving like a siphonophore.
Yep.
Yep.
that's the same
We can
one.
keep
Yeah.
cruising. So naming species, I mean, J-Rod does it all the time. Those are common names. They're in English. They're really useful because they're easier to communicate, easier to pronounce. We can share them with people who aren't versed in biological Latin. But official names, so each species, each officially described species gets exactly one name that's usually Latin or Greek in root. And to do that, you have to write a paper with all kinds of measurements and drawings and probably photographs, and you've got to put a pickled specimen in a museum somewhere. And it's quite a process. It's good though because it leaves absolutely no ambiguity as to what species that is, whatever species you've described. So to answer your question, if we wanted to officially name a new species, anybody here could do it, provided they were willing to put in that amount of work. And provided we could recover a good enough sample. Oop, that is an amber lobate. Let's get a little bit of video and then try to slurp it. Good spot.
Working with that tether, but got it.
Mm-hmm. There are also some interesting rules about naming species. For instance, it is severely frowned upon to name one after yourself. That was a thing once, but it doesn't happen anymore.
I guess that's not gonna work for me then. I can't name it after myself.
You can get things named after you.
Yes, yes. Maybe the only reason you should be nice to us, Ben.
Yeah, that's true.
Let it get back into its form.
J-Rod's just squaring up on this lobate to try to get it into the science camera. No pressure. These deepwater lobates are a perfect example of midwater animals that are intolerant both of high oxygen, and most of them also can't handle low pressure. This one's probably kind of on the boundary of where it would be able to survive depressurization. Hopefully it'll come up in okay shape.
Am I good to sample?
You're good to sample. Thank you.
No, nothing back. Or you gotta go forward.
Move him back, I gotta get your guys on.
Bridge, updated position.
Copy, Ben.
Ooh. Yeah. Whoa, nice.
Clear?
Yep. Why do we choose to sample this one? Good question. So this is obviously a midwater pelagic ctenophore. These are animals that never really see a hard surface in their lives. And yet, despite that they're drifting apparently freely through the water column and through the major ocean currents, we do see structure among the populations. And what I mean by that is that we find different species in different locations, just like you would expect from species on land. Say, for instance, the birds you find in eastern North America are different than the birds you find in western North America. And that's pretty intuitive because there's mountain ranges and stuff in between, keeping them from interbreeding. But we don't really know what keeps different populations of midwater organisms from interbreeding. Is it the structure of the currents? Is it the water temperature? Is it underwater mountain ranges? These all remain open questions that we've just started to answer. So this particular species of comb jelly is interesting because it looks an awful lot like a species that we see much further north in Central and Southern California. But we won't know whether it's really the same or whether those populations interbreed until we sequence some of its DNA, and that's what we plan to do with it.
Still hasn't come through yet. We had a really low section, so I can try to speed it up a little bit.
You can kick it up a smidge if you want.
Either that or it was so wispy when it came through.
It just came apart. I still think you'd see something. There it comes.
You see it?
Yeah. I wish I were more confident.
I don't see anything.
No, it can't be it. Well,
See my hose in the background? It's off around. And I put another kid,
yeah, it got pretty whipped around.
but
Did you just turn section off?
Yeah. Just see if like anything were to slow down from
Yeah.
the screen.
Yeah, they do get screens sometimes. In fact, I wouldn't be surprised if that guy would just go through the screen. Eh. You win some, you lose some. Yeah. The alignment looks right. Oh well, we'll find another.
Just
Maybe we'll get a twofer.
bringing it on fast and then slow.
Yeah, I mean, if it's already hit the screen, then we're kind of done, but.
How? Huh, strange. Definitely went in, right?
Yeah, it definitely went in. Unambiguous. Technically, the permits where we are issued by the Ecuadorian government because we're in Ecuador's exclusive economic zone, or EEZ, which extends 200 nautical miles from the coast. Earlier, when we were diving iguanas and pinguinos, we were inside of Ecuador's Galapagos National Park, so that has even more stringent regulations and permit requirements. We'll be bringing all of these samples ashore in the national park in Puerto Ayora on Isla Santa Cruz in the Galapagos, so we're basically required to meet all of their scientific sampling requirements. Yeah. Mystery.
Talking about do they get pretty big?
I'd say that was a, like top 80th percentile individual, at least based on the ones I've seen in California. I mean, the ones out here could be totally different. And that amber one, as far as I or any of the people I work with know, is an undescribed species.
Mm-hmm.
But if you want kind of a reference point, it probably belongs in the same genus as the bloody belly ctenophore, or Lamont-Doherty. That's the one I gave you the sticker of. You can find a lot of pictures of those online. Yeah, we're very much indebted to the folks with the Ecuadorian government and some of our Ecuadorian colleagues here. We have a couple of observers aboard. There's one scientist from the Charles Darwin Foundation, which is sort of responsible for stewardship of the Galapagos Islands. works closely with the government. And then Ricardo and Denise are from the Galapagos National Park and the Ecuadorian Navy, respectively. So it's pretty cool to have them along. Now, a lot of things in the ocean are linked to the moon. Certain shallow water organisms spawn by moonlight. I don't really know how deep those moonlight wavelengths reach. People have studied it, I just don't know off the top of my head. Tides are obviously a very real influence of the moon. And we actually see tides measurably at hydrothermal vents. They're not unlike tides in tide pools. The difference is that instead of the water level coming up and down, the level of hydrothermal fluid goes up and down. That's a siphonophore. I think it's a little baby Irena.
The bottle brush one that we were
Yep,
talking about?
pretty sure. Just a small one.
Keep going?
Yeah, let's keep going. I'd say the bookkeeping requirements for sampling are stringent. I can't blame the Ecuadorian government for that. Everyone we've worked with has been really great, reasonable, and a pleasure to work with. Yeah. Yeah, so hydrothermal vent tides are they're similar to tide pool tides, but actually they're opposite, because when the tide at the surface of the ocean is high, the additional amount of water weighing down on the vent site essentially slows down the flow of fluids from the vents. So it kind of pushes the fluid into the vents more, and the flow decreases. When the tide is low on the surface, the water is actually a tiny bit shallower, and the flows get measurably faster and hotter. Maybe a halocreatid? Yeah.
Yeah, the little crown one?
Yep. That's well, I mean, it looks like a crown, but it's confusing because there's a group called coronate medusae, and this is not one. This belongs to the Halocreatidae family. It's a Hydromedusa.
Sounds like a coral.
Yeah.
The end of what you're saying there?
Well, so the Idae is a family thing, basically. Idae or Inae, they all pretty much end that way. But yeah, this is why we need people like you making up intelligible names, J-Rod. All I request is that this one doesn't have crown in it, even though it actually looks a lot like one. This might be a Halocera, might be the species name. I wish I had a common name for it, but you all are invited to submit suggestions.
It does look like a wheel spoke in the top.
Mm-hmm. Yeah. Too bad pinwheel jelly is taken. Kind of looks like a basketball. Kind of looks like the well, yeah, that's a stretch. They don't meet in the right way. I think that might have been the same thing.
index away from 2, just in case there's any?
Yeah, I think that's a good idea. Even just a little bit we would be able to use. Thanks so much.
For you.
No.
Bread treats.
Too
There's no
thoughtful.
banana bread, so I just
Thank you.
put anything else.
This is perfect. Thanks so much. Mm.
How did you like your coffee tip?
Anyway, caffeinated. Is that another one of those tiny little sipes?
Kind of seems like it.
I think so.
I was flying by it.
Looks really, really similar to the one we saw earlier.
It's like a little puffball. So I thought it was.
Oh, that's not a sipe at all. That's a cluster of radiolaria.
Oh, yeah.
Yeah.
Okay.
So we call them, Mbari slang is cluster rad.
Cluster rad.
It's pretty rad. 20 of them. So these are small.
Super
Well,
tiny.
I suppose they're small for animals, but big for protists. Large sort of colonial protists that make their skeleton out of silica. And whenever you guys get a proper microscope on this vehicle, we'll be able to see these sort of intricate geodesic snowflakey patterns
Yeah,
that
same right there.
form. This is pretty good. I'm not complaining yet.
Wow.
Yeah, basically up really close, closer than we can get with an ROV. The sort of inner sphere inside the spikes looks like a giant geodesic dome with a lot of tiny triangles. Anyways, they catch schmutz falling through the water column, and that's what that cloud was around them. The structure at the center is the organism itself. So they belong to or at least they've been reclassified, but they used to belong to a group called radiolaria, which I think refers to the kind of radial, round, spherical structure of the organism. A really fascinating group. And a lot of them also luminesce.
Oh, cool. So where's the which way
I don't-
camera now?
I don't think radiolarian, we've tried to pick up radiolarian luminescence, and I don't think we've ever succeeded with the ROV. There's still time to put that A7S into that housing of yours, J-Rod. Yeah, cran-cheated squid. They're always cute. Yeah, the lifestyle of a radiolarian, or phaidarian is the new proper classification, is kind of similar to a larvation. You know, they cast a mucus web and they just pick up detritus that's sinking through the water column. They're not related, but like a lot of things in the deep sea, they've convergently evolved ways of making a living. Yeah? That's your final answer? I don't know if the polka dots are built in or they're reconfigurable. That's a cutie. What are they called? I think piglet squid is the name I've heard applied to these.
Yeah, I've just never seen one moving that fast, but yep, you're right. Now
Yeah.
that it's slowing down and doing that cool move.
Actually, larvations in the family in the group Fritillariidae tend to they naturally have kind of a greenish yellow cast. So something could have been up. There could have been a different color to the particulate it was picking up, or it could have just been the natural color. There we are. Yeah, that's the one. probably Helicocrantia. Oh, there it is. Now it's doing the pose. My sister calls these Moomin squids, for anyone who's a fan of Tove Jansson's cartoons. Times 4.
Thought you were going to. Isn't it right now?
What's that?
The meeting's right now. He told me 7 minutes. I won't have it. You like it?
Are we being rushed?
No.
Should I? Okay.
No, no, no. This is a meeting that Carl was trying to get me to.
I agree, they don't look like part of the organism. To me, they just kind of look like marine snow, but they could very well be amphipods or some kind of parasite, commensal. We do have to keep up with our winch and our schedule here, so while J-Rod does a really good job of getting good macro footage of some of these animals, we can't hang on them forever and look at their natural history.
Yeah, it's really tough when it's like the areas where it's like bang, bang, bang. Good, good, good.
Yeah,
Okay, tap
right.
back up.
Oh.
Looks like a depleted foundation.
Oh, I didn't spot that. I saw the blackfish in the foreground.
Whether that looks like they're fully done.
No, there's no way.
I don't see them. I can get them out in the distance and try to sneak up on them. I haven't seen any snake bills yet, huh?
I don't remember. I think we saw one up off the bottom, I think. On one, like on pinguinos, maybe, that I was a little bit surprised about. But like one, not, normally they're pretty abundant. Normally, I say, having never been here before.
No, there
Oh,
was one.
yep.
As I just
Speak of the devil.
They, a couple of cruises ago, one of the guys got some pretty good images of one of those, looper reels?
Gulper.
Gulper reels? Like,
Yeah.
it did like the big mouth.
With the, unfolded its pharynx.
Amazing.
Yeah. And Bari spends way too much time with those guys because they're the mascot, they're the logo. So they're always looking for better footage. Yeah, thinking like, where? Okay, let's check it out.
The way it tapered at
I
the
like
end.
your attitude. No, it's a sinker.
Okay. But the way it tapered.
Yeah,
looked like
I see it.
Looking out for you.
Appreciate it. Solmicis.
didn't know how to spell them for the longest time, so I would label the pictures the letter T.
There you go.
Space N-O.
Mm-hmm. I'm sure that license plate is taken, but I wish it wasn't. That little jelly thing. It was interesting.
Two tentacles, or?
Well, I have a feeling it has 4, and it's just in a position to make it look like it has 2, but let's look. Oh,
I think you're right.
I think it, well, now it has 3, but it might have had more at one time. There's a little, yeah, it's like, I think it's Solmaris or something down there. Yep. Little red jelly. Not sure who.
Right, keep going.
Yeah, we can keep going. Oh, good, you're back.
140 meters, they were at the 500 stopping zone. Yes. 50-meter vessel moved out to the northeast.
50 northeast.
Yeah, let's see the way it curves pretty much. That push me. Just take the dot, go to the 50-meter zone.
We're definitely. It's a shrimp. We're in the DSL now. Everything's moving down. You can see the shrimp. Everything's. Anyway, sorry. Oh.
Pretty jarby.
DSL.
Deep scattering layer.
RV bridge.
Got you an updated waypoint.
Gilby, thank you.
You say they tend to be more common in the oxygen minimum zones?
The lobates? Yeah, these ones, yes. The red ones and the amber ones generally. That's an atolla. That's George Matsumoto's baby. We don't have a permit for them, but I'm sure he will love the video. And maybe he's watching right now. So, hey, George. Anything special about this one? They put on a brilliant bioluminescent display also.
Oh, they did?
Yeah.
Why did it have the one long?
You should ask it or ask George, but they all have that. That's how they feed. I think they're snow eaters. I think they pick it up on that, but I don't want to get that wrong. It's a characteristic, though, of atolla. They all have that long trailing tentacle.
Is it something I can retract?
I don't think so. I do think it's something that breaks off. You probably regrow.
About to touch it.
Beautiful.
If you're watching.
Well, if so, he hasn't piped up.
I think they've got a seminar today. I don't know if he's.
Yeah, he's probably got plenty to do.
About to get things off. Very good. Takeaway shot. Buddy.
Yeah, that one's really giving you a show. But
Yeah.
we have schedule to keep. Yeah, the deep scattering layer. I mean, you said you have a Navy background, so I think it was sort of classically known as the false bottom. When the DSL gets really thick, all the air bladders and the fishes and stuff, can give the appearance of a raised bottom.
I was in the Navy for 8 years, but never once was I on a boat. I did aviation. I flew on a plane that was too big to land on. And then I got a job on the boat.
In part the time of day. So it's predominantly made up of diel vertical migrators. You know, we love to throw our three-letter acronyms around. And a couple that you'll hear are DVM, diel vertical migration, and DSL, the deep scattering layer. Basically, the deep scattering layer moves up and down.
And that's just where the majority of those particular animals are.
Yeah, I mean, what's thought to drive it is they make the migration to the surface, to the more nutrient-rich waters to feed at night when it's dark and you're at less risk of being eaten by things that can see you.
Right.
So I think a lot of factors go into it, and it's been extensively studied. It's just not my specialty, but things like you know, turbidity, the amount of downwelling light from the surface, obviously time of day on a finer scale.
It's a big cephalopod.
That is a big siphonophore. I think apolemia. That's okay. Keep going. So
Biggest migration on the planet, and it happens every day. I guess it's consistent, constantly happening all day long.
pretty much. A lot of things basically turn around as soon as they get there. And the crazy thing is we know little to nothing about the biochemistry of migrators. Like how much do they have to alter their cell fluids, membranes, things like that every time they make the trip?
That hasn't been studied.
No, not really. Not that I'm aware of.
Is it particularly challenging?
I've certainly looked. Well, it is challenging because you have to sample things on a fine time scale. You either have to run ROV dives that like follow the DVM and or come out of the water twice a day. Oh, there's a Bathosauroid. We're interested in that. Or you need to like do trawls day and night.
I mean, is it something that you could simulate in a pressure chamber?
You could, but I wouldn't be confident. You could, and I've actually discussed it with some people. But it would be a technological heavy lift. It would be pretty cool. Pretty cool sort of incubator.
This is not the amber one. Right?
No, but it's still of interest. Yeah, we call this Bathosauroid, or just the deep sea lobate. It also belongs to what we call a cryptic species complex, which means there are a bunch of them. We know genetically that they're different species, but they're really hard to tell apart by eye.
And this is a sample?
This is a sample if you can get it. Yeah.
That's good right now.
I have some samples in the bank, like that in the freezer that I'm hoping to analyze that are from one of Vanilla Choi's cruises where we were trawling like 2 or more times a day.
You said you collected these at a trough?
Hmm?
You collected these at a trough?
No, those were shrimp from her. Yeah, these are not trawlable.
That's what I asked.
Yeah. Yeah, if we could do day and night collections of these, we'd love to, but obviously there are reasons we can't do that in Monterey Bay.
Got it.
Nice, thank you.
And indexed. So, that's
What was this ctenophore called?
You can just call it a lobate, or for extra credit, Batho, Batho, Cyroe, Cyroe. Yeah, that works. That's a common name that I made up. It's not very exciting, but Batho is, Batho means deep. It's part of the name, so.
That's us at 500. Can we stop here?
You don't need to stop, you just need to power on super.
Roger that. You got it?
Yeah, super start, or turn on rather. We're just power, like cycling it.
Alright. Super has power.
Thank you.
Is there a specific filter?
We're not doing a filter, so you can just write regular observation of just testing. Even
Nice save.
you don't
It's
even need to
okay.
do super start, because you can just go a regular observation. So we're just testing with the comms.
just to keep everyone up to date, we are on the way up from the Navidad vent field on the Western Galapagos Spreading Center. We're having some fun in the midwater. J-Rod has just found us another lobate ctenophore that I'm very curious to see. And I don't think we'll want to sample that one.
You know?
No. It's pretty though. Maybe we'll get a glimpse of it on the science camera. Maybe not. Yep. So this is a lobate ctenophore of genus Bolenopsis. These ones compare to the last one, Bathosauroi, that we saw in kind of an interesting way. Their ranges of depth overlap by a couple of thousand meters, but in most parts of the world you can find Bolenopsis all the way up to the surface, like you can snorkel for them. But you will never see Bathosauroi within about 200 meters of the surface. A big part of my PhD thesis was looking into the biochemistry that causes that to be the case.
What is that?
Whoop. It's the cell membranes, best we can tell. Yeah, I have some micrographs that I can show you of the membranes of Bolenopsis and Bathosauroi, and basically what you can see is as soon as you take Bathosauroi like a couple of degrees, or that's the equivalent, biochemically speaking, biophysically speaking, of a couple hundred meters above its sort of native range, the membranes just turn inside out, and the cells turn into a bunch of tiny bubbles. They split into little tiny compartments, which obviously
Doesn't work.
it's a bad day. Yeah.
Deck control, comms check.
Can we get you item clear?
Up north, 400 meters is the magic depth for lobates. That's when it all happens. Don't know what it's like here, though. It's pretty cool to have been able to be in here through the entire ascent. Get a real survey of the water column. Fish. Solmissus. There's another one in the background. But as J-Rod was pointing out earlier, this Hydromedusa solmissus is probably the most common, larger than a fist thing we see out here. That one is different. Yep. That's a coronate. I don't know exactly who that is. Solmandella maybe, but I'm just guessing. Okay, we have a request from Nicole on the biology team to, I think, make some tracks.
We haven't stopped. I mean, we stopped a couple of times.
Mm-hmm. I'm going to clarify with her.
Everybody thinks that we are stopping all the time. Just because we're looking at it. The only times we've been stopping is actually just for the samples.
Okay.
And even that, it's a short stop.
Oh, got it. Okay, she just said when the RV comes up on deck, she just wants to get at it quickly. That's all. We're warm and dry with our coffee mugs topside. I'm not aware of any human-occupied vehicles that could have been down for a fifth as long as we just were. Are there any? Anything with a comparable endurance? I mean, aside from military samples. Research vehicles, excuse me. Who is that? That's a little scythe. It's a weird one. Yeah.
What's that?
What depths do you put on the surface?
ROV depth 100. Just it depends on how far I'm strung out. That's another one of those sea
Cucumber, yeah. Pelagothuria.
You'd like to get what?
I would like to choose the one stream at 100 and get all of the depths.
Yep. Whenever you want, Fade.
Yeah, 100 is fine.
Okay.
150 or something.
All right. Didn't spot a phronema, but I wouldn't be surprised if somebody out there has sharper eyes. We are in our last 171 meters of streamed ascent at a rate of about 20 meters, average of a little bit more. Yeah, yeah. I think you think we're averaging a real 20 meters a minute.
24.
Okay.
That's
So.
consistent.
Yeah.
My rate is.
Yeah.
If you see where it says winch.
I see what you mean. Yes.
That one's going to stay at 24. If I'm going to outrun it for a little bit, I'll stop. My delta will get small.
Understood. So that gives us 7 more precious minutes with you exploring the midwater on the Western Galapagos Spreading Center. Oh, that's a pretty common ctenophore, though, you know, it would be nice to have one. We'll see a bunch more, almost guaranteed. Yeah, see, there's another one.
To the right.
Yeah.
To the right.
Yep. Those are super hardy. I can slurp them at full power for all I care. So this is a sidipid ctenophore, commonly referred to as a sea gooseberry. Probably belongs to genus Hormifera, which if you translate it means something like bringer of excitement. I'm on my 3rd coffee now, so it's probably going to be an accurate name. They're one of the few genera of ctenophores that do not create their own light. Here comes a bridge.
Contract.
Welcome, sir.
But you can see the ROV lights bouncing off. I wasn't serious about full power, but it's fine. Just kidding. Oh. You actually could slurp it at full power. It would be fine. It will be fine.
I slowed down. He's in there.
There it is. Yeah, it's there.
Index.
Indexing. Thanks.
And did stop the winch.
Hormifera 1427. 52.
I've been writing this down. What did we put in 3?
Bathos-Airoi.
2 is the one that never.
Oh,
came
so
through.
nothing, we put nothing in 2?
Well,
2 is a question mark.
big question.
I'm happy to keep my own books too. Don't worry too much about it.
Okay, because I had the Bathos-Airoi ctenophore SS2.
Oh, okay. That's SS3.
That's actually SS3?
That's actually SS3.
Okay.
Yep. Oh, that was an interesting looking one.
Then
That's
was
okay, it's gone.
the
It's jumping.
Pteroides ctenophore in SS1?
Correct.
So, wait, I was here for SS2, so what happened?
Yeah, we sucked one of those amber lobates, and it just never really came out the other end.
Wait.
We never.
I didn't see it though.
Well, maybe you were paying attention to more important things. I don't know. It's
Okay.
been a long night.
Is this SS4 then, or SS5?
This is SS4.
SS4.
Yeah. Ooh, that's an interesting ctenophore.
Is that
It's
next?
down center.
Yeah.
I don't know what it is yet. Right there. If you have the leash to do it.
For a quick one, yes.
Yeah.
And then if you want, then we can
Yeah, let's sample it. I would just say go ahead if you think you can
go
get
ahead
it.
and sample.
Yep. Yep. Undescribed mertensid ctenophore.
What is this?
Well, it's a mertensiid, MERTENSIID.
It's ctenophore type.
Yeah, Ctenoctime. Not only are these probably a cryptic species complex, but they don't even have a name. caught you. It's reasonably hardy too, this one.
Try not to mess them up.
That's okay. It's a
Yeah.
yeah, we are just beyond the mesophotic zone. Still pretty dark here. Visible light tends to reach to about 200 meters in these clearer parts of the ocean. Wow. Nicely done. Sweet. Sight.
Yeah.
Thank you. Keep thinking one of the chief scientists is going to come in here and bust us both. Yep.
Take that.
Yeah, I know.
At least it didn't seem like that took over.
I heard you say payout though. I heard you say payout though.
Yeah, because I was sitting at 17 when we were looking at it, so I just needed a few more to make that collection go a little bit quicker.
How fast? Because I know this is going to be a long ROV turnaround, right? Because we're tow-yoing. When do you want the suction samplers
Yes,
back?
we put in a note that the super starting in
Right,
Just
go
'cause-
forward.
Cool. Because I'm committed to helping out the muscle team and some other people, so I'll probably park it
Yeah,
in the
right
cold
down
room
in
for a little while.
Like at 300 meters depth, it started misbehaving.
Yeah,
Yeah.
it's acting up again.
Yep,
Super start that up?
yep.
Okay.
It was working from 500 to 300.
Shallows.
Yeah, that ctenophore is another undescribed species. Surprise, surprise. And we would love to know about its genetics, which is why we collected it. Those little branches on the tentacles are called tentilla, and a lot of people theorize that they mimic little crustaceans that live in the water column and look tasty to fishes and stuff. All right.
We still got another 160.
Yep. Thank you all for joining us. Fight, who's been adeptly running our stream from the control room, has got some other duties to attend to today. So we're going to sign off for now. Dive 570, broadcasting from Falcor 2 on the Western Galapagos Spreading Center. We hope you'll join us again for our next dive soon.
Relaunch. Then relaunch, yep.
Is that the stream over?
Not yet.